Hello, my name is Marc Lipman and I'm Director of UCL-TB, and on behalf of my co-hosts, Finn McQuaid and Toyin Togun, who are directors of the London School of Hygiene TB Centre, I'd like to welcome you to World TB Day. As you know, this celebrates the fact that 140 years ago, Robert Koch publicly announced the discovery of the cause of tuberculosis. We celebrate that with a joint symposium, which has been running now for several years, where we look at what is happening in tuberculosis through the lens and the work and the research that we perform here in our laboratories and clinical practice. This year, as you know, we made the decision to have a virtual meeting, and I'm delighted to say that we have across the world over 750 people who've registered for this event. That's great news. What's important is we ensure that you enjoy it. And I'll take you through some of the features that we've built into this meeting. What I also wanted to say is summarized in terms of the agenda that we have. Next slide. As you can see, we're trying to cover a variety of areas across tuberculosis. Central to this is the patient. And I'm delighted to say that we have, as our first presentation, a video recorded of a patient's experiences of TB and the consequences. We will then progress through the afternoon, looking at understanding M. tuberculosis in the post-genomic area. And following a short break, delighted to present new information on pediatric tuberculosis and the adoption of clinical trial data into WHO policy and hopefully practice. We can't avoid COVID-19 at the moment, and we have a session that's focusing on that as one of the other great pandemics of this current century. We're then very delighted to say that we will then be handing over to UCSF, and I will give you more details of how we would be doing that in our next slide. That final session covers both us and UCSF, who, as you know, are 8 hours behind us. So that is the beginning of their day. A few housekeeping rules for you here. The way that we've structured it to maximize impact, we hope, is that we will have the videos playing of all the speakers at the start of each session. And then throughout, please put questions for panelists into the Q&A that you'll see at the bottom of your screen. But if you want to actually communicate with other members of the audience, then put those into the chat. Do please let the rest of the world know about this session, and also note that these sessions, if you miss any of them, they will be available later. What we will do at the end of the videos is we'll have between 15 and 20 minutes of hosted and facilitated conversation. So for example, if you've put a question to a panelist, the chairs of that session will then pick that up and bring that out. So they will be asking the questions on your behalf. The final bit, and we'll go through this again later as well, when we link up with UCSF, as you can see, there will be a separate way of contacting, of linking to them. And that has been emailed to you, but will also be made widely available. I do encourage you to join what looks like a really excellent meeting, and certainly the first session where we have speakers from London and our centres presenting. Next slide. It now gives me great pleasure to ask Professors Ibrahim Abubakar and Professor Liam Smeeth to give the formal introductions on behalf of UCL and then the London School of Hygiene. Ibrahim.
Thank you very much, Marc, and an absolute pleasure to be here. So it gives me great delight to speak at this year's World TB Day event. For those that don't know me, my name is Ibrahim Abubakar. I'm a professor of infectious disease epidemiology at UCL, and I lead UCL's Faculty of Population Health Sciences, which brings together 8 of our institutes: the Great Ormond Institute of Child Health, the Elizabeth Garrett Anderson Institute of Women's Health, as well as the departments responsible for cardiovascular sciences, global health epidemiology, health informatics, clinical trials, and our new global business school. So I get asked often to give opening remarks in a number of events that we run. But this one is particularly special to me for a number of reasons. And it's really an honor to come back to home, essentially TB, to speak. I have worked in TB for the best part of the last 2 decades, and it is there to me because TB epitomizes all that is wrong and all that is possible about humanity. We all know that it disproportionately affects the poor people, the poorest in society, but also it has the potential to affect any one of us. It's a largely preventable ailment, which sadly continues to afflict millions of people and cause 1.5 million deaths. And we all recognize that it is possible to rid mankind of this scourge, and yet we fail to do so. So a great deal of this failure is due to a paucity of resources dedicated to TB research, TB innovation, and TB programs. Having had the opportunity to chair WHO strategic and technical advisory group for 5 years, to lead England's TB unit in Colindale, and to work as a TB academic for the last 2 decades, and work in low, middle, and high income settings, I believe that the theme of this year's World TB Day, Invest to End, saves lives. I believe that it's the right one and it's apt, because that's exactly what we are getting wrong. We are not investing enough in TB, both research and in innovation. I'll therefore like the WHO and the TB community in asking our political leaders to respond to this call and invest in TB research and control. However, I'd like to point out that I do think when we call for TB research funding, we need to always remind ourselves about funding the entire health system. But then well-funded health systems must have provision for TB research, TB prevention, TB diagnostics, and treatment of drug-resistant and sensitive TB. And it's only through doing both that we can ultimately control TB. TB killing 1.5 million people annually is really a sad state. A lot of the progress we've made over the last 2 decades has largely been unraveled by COVID-19. We are failing to detect cases, and too many people are dying because they have not been able to access care over the last 2 years. It is therefore puzzling to me why anyone would actually debate the need to put the right resources in TB control. Our world is currently beset by multiple crises, not least the COVID-19 pandemic stretching national economies and therefore making it challenging to find any new resources to support TB control. The war in Ukraine is resulting in untold suffering and hardship to the population of the country, but would impact the rest of the world socio-economically, again, making the call really difficult to find the resources. But in addition, Ukraine has a high burden of TB and MDR TB, so individuals with TB in that setting will also suffer a side effect directly. This war would impact the global economy in ways that we cannot predict and lead to potentially catastrophic expenditure when people develop TB in multiple countries. Nevertheless, for me, the lesson of the COVID-19 pandemic is that if you put in the right resources at the right time to the right problem, it is possible to solve it. And I'm delighted that major funders are looking at possibilities to support TB innovation and vaccine research. And I think we should continue to advocate for that, and we must do more. We need to increase the funding for both preventive therapy, early case detection, and ensuring that everyone with TB has access to treatment. I am delighted to see the range of TB science talks being advanced by LSHTM and UCL together today, and partners in the Global South to address some of these major challenges. And I look forward to the interesting presentations today and to learning from the contributions that we are making to make a difference globally. I hope you have a very productive session, and I look forward to the activities to commemorate the day. Thank you.
Thank you so much, Ibrahim. And now if I could call upon you, Liam.
Yes, indeed. Hopefully you can see me. I'm Liam Smeeth, the Director of London School of Hygiene and Tropical Medicine since August last year. And I'm delighted to see this partnership event. When I'll go back a long time to the late 1980s, I was going to say 1880s, when I was at medical school and in late 1980s. And there was a feeling that TB was a kind of old disease that was dying out. You know, more exciting things to study, there were more for the future. And of course, that hasn't happened. In fact, it's continued as a huge challenge, as it in fact has emerged as an even more major challenge with many factors, including HIV contributing to that. And as Ibrahim said so well, the impact of TB is so focused on the most disadvantaged people with the least resilience, and I think that's both within countries and between countries. And what, I guess, an overview from me as a sort of non-specialist would be there's clearly so many complex factors involved in what's driving TB around the world. And there isn't going to be a single solution. So I probably sadly have to say to everyone here, you're doing fantastic work and you'll achieve huge amounts in your careers, but you're very unlikely to discover the single one thing that gets rid of TB forever. But that lack of a sort of magic bullet, don't let it get you down, because what it means is there are probably thousands of factors contributing to the continuation and spread of TB and all the damage it causes. And because there are thousands of factors involved, it means there are thousands of ways that we can tackle it, some of which we know about and some of which we don't. But we really need to carry on pushing on all fronts. There isn't going to be a single solution. And this means we really do need a multidisciplinary approach, right from molecular approaches and diagnostics through to social sciences and economics across the whole spectrum. It's very clear with something like TB that the solutions and what will help in each setting and each population will be very context specific. So we're going to need multidisciplinary partnerships, we're going to need geographically mixed partnerships, and really coming together in a collaborative approach. And I think we've seen from COVID where collaboration has really helped and where lack of collaboration and lack of partnership has led to isolation and much worsening for many populations. So it's really great to see this meeting happening in partnership. There's a clear need for continued work, in fact, increased focus to tackle TB worldwide. It comes back to the basics, what we need, and what I think the people attending today and your colleagues can do is deliver the evidence to guide policy and practice and train and encourage that next generation of health scientists. And they really are the linchpins of how we'll make progress on a complex problem like TB. So program looks fantastic. I'm not going to be able to stick around for long, but I wish you all good luck with today and all your work. And it's great to see this partnership working well. Thank you.
Thank you so much, Liam and Ibrahim, for your fantastic words of support and encouragement. And now we move to the formal first session of UCL-TB. Next slide, please. And if I could welcome my co-chair, Katharina Kranzer, for our first session, Health Consequences Following TB. What I'm going to do is introduce the first video, and then Katharina will pick up to take us through the further 3 videos. And then, as I say, please post your questions in the Q&A. Katharina and I will then feed them to the live Q&A panel. So it gives me great pleasure to thank Hanif Esmail for interviewing a patient, Rahima, who kindly agreed to discuss her experiences of life both during and after tuberculosis. Hanif, I should say, is a clinical associate professor at UCL and at UCLH, who is also part of the key North Central London TB Services Network team based at the Whittington Hospital. Thank you. Thank you. Very powerful and really something to reflect on as we spend the rest of the session now picking up on the other videos. So if I could call on Katharina now to introduce Dr. Matt Quaife.
Thank you very much, and thank you very much also for this very humbling first talk and interview. It's a pleasure to introduce a lot of colleagues and friends in this session. So Matt is a health economist who recently left his assistant professor position and has now taken up a consultancy position, but he is going to talk about his and other people's work on post-TB modeling. And just to let you know, please put your questions into the chat or into the Q&A. And Ryan Huben is on the panel and will take any questions about this talk specifically later. Thank you very much.
Hi, my name's Matthew Quaife. I'm a health economist based in the TB modelling group at LSHTM. I'm going to talk today about a hidden epidemic, but for how much longer. Talk about post-TB modelling economics, and I'm presenting here on behalf of a number of colleagues, one of whom, Ryan Huben, will be participating in the Q&A after this talk. We're going to briefly outline what we have known before about post-TB and sequelae. I'm going to highlight some recent work and advances in post-TB modelling and economics. I'm going to highlight some key questions and next steps for post-TB research. So up until recently, we've had very strong, but nevertheless anecdotal evidence that people do not recover to full health the minute that TB treatment stops, which is what many of our models have assumed. The work I'm going to outline today is demonstrated in this diagram, where we have someone living their life on the x-axis. We have the extent to which they incur disability weights in the DALY framework on the y-axis. They're at full health before they get TB, their health drops. And at the moment, we assume that their health goes back up to full health and they live for a normal life expectancy. What I'm going to talk about today is work to estimate this kind of bluey purple area to get understanding the morbidity and mortality from post-TB disease. First on mortality, Kamila Romanovsky conducted a meta-analysis published in 2019 that suggested a standardized mortality ratio of just under 2.91 for people with TB compared to a control group. I don't work much with standardized mortality ratios, but I'm told this is a huge one because it considers kind of analogous to the COVID epidemic of all-cause mortality. This is a huge increase for people with TB who have had TB compared to a control group. We took this work initially and estimated how the burden of TB in India would change if post-TB DALYs, that's mortality and morbidity, were included. On a bit of a back-of-the-envelope calculation estimated that DALYs would increase by 54%. If we included post-TB DALYs. More recent work by Pete Dodd at Sheffield and colleagues have estimated that there are 155 million TB survivors alive today. You may be surprised that we need a model to work out that number, but it's critical for planning post-TB care to know how many people are alive with TB. Other insights from this paper is the global the global distribution of people who are alive after completing TB treatment, and the ratio of treated or people post-TB who are treated and untreated across different ages, across different areas. We see here familiar patterns across the different geographies and age groups. Pete's work also estimated that there's a really big number here, 3.5 billion life years lived post-TB in the 40-year period between 1980 and 2020, and there's 27 million people were treated in the last 5 years. So this is really important data to understand quite how many people are potentially facing post-TB mortality and morbidity effects. Work with Nick Menzies, which some colleagues at LSHTM and myself were involved with, estimated disability-adjusted life year losses, so DALY losses, which measure mortality and morbidity due to post-TB. Nick's primary results are on this slide, and what we see in the figure on the left, the pink bits of the bar represent DALYs lost, so mortality and morbidity due to TB disease. The light blue parts on top are DALYs lost, again, mortality and morbidity due to post-TB sequelae. So when people are recovered from TB infection and have completed a course of treatment in many cases, these are longer term impacts of mortality and morbidity. On the right, we see that the life years lost, years of lost life, increased by 63%. So that's our mortality effect, increasing by 63%. But really notably here, years of life lived with disability increased by 341%. So a huge increase in morbidity when we consider the implications of post-TB DALYs. This is particularly interesting because when we normally do DALY calculations for things like TB, we see huge mortality effects of impacts on life expectancy. But because TB at the moment, we only assume TB disease lasts for 2 years or so, the disability impact is quite small. But considering these longer term impact on disabilities, we see huge increases here. And overall, we estimate that the number of DALYs would increase by 80%. So almost, we could say almost doubling perhaps, if we include post-TB alongside TB disease. So moving on to what we need to do next, there are some key questions and next steps for post-TB research. First of all, Sedona Sweeney and others are working on ideas around the economic impacts of post-TB. Now we have this framework shown below, which looks at kind of in a traditional patient cost framework of how the impact of TB impacts the household economic situation. We know that there are huge costs before TB diagnosis. We know that there are huge costs during TB treatment, and it's highly likely that this missing piece of the puzzle is also going to result in significant economic impacts of post-TB, but we just don't know what they are yet. So Sedona and others are working with cohort studies and doing some modelling to estimate the total economic impact of post-TB. So just to summarise the key questions and some next steps for post-TB research, I think we know now the burden of TB, the burden of TB and the burden of post-TB is demonstrably massive. That can be impacts to the individual, to health systems, and at the global level. And there's a joint epidemiological and economic burden that we need to understand and deal with. There's a lot of variation between groups that we need to understand better, for example, between individuals of different ages or between different countries and regions. And as with lots of things around TB, causation is really difficult. Lots of things are correlated with TB infection, and we'd expect lots of things also to be correlated with more severe or less severe post-TB sequelae. A few next steps for strengthening our data that we have on post-TB. So this is my call to measure what we need to be measuring. We need a strong and comprehensive definition of post-TB because it's hard to measure what we can't define well. We need to think about if we can use routine data to better understand things that correlate with post-TB and whether things get worse or better with different correlates. It's really difficult to imagine an RCT doing something around, we can't assign people to have post-TB burden or not. So we need to do more with these data that we have, perhaps with innovative study designs or analysis strategies. And we also need to measure more. We need to capture and understand the full range of economic and health harms in the short and long term. On the modelling side, thinking about epidemiology, modelling and economics, we need to move away from modelling for advocacy to start modelling for solutions. So I think a nice analogy is from the HIV world, where this is a pretty famous, I think famous is the word, Greenwich paper, which showed the impact on HIV incidence of universal voluntary HIV testing and immediate ART. So for those of you who were around before, big trials doing this paper really stimulated discussion of, oh, let's get to this green line, let's universally test and treat everybody. And that led to real world trials, the pop-art trial, of which results were published a couple of years ago, and I'm sure investigators from that trial will be listening today. But the point here is that this modelling spurred on trialling and the epidemiological studies to investigate the what-ifs. So therefore, I think we need to investigate how including this post-TB mortality and morbidity will change our priorities within treatment programs. For example, we're doing modelling, look at the cost effectiveness and impact of a vaccine. If a vaccine is averting lots of post-TB DALYs, that vaccine is going to be a much more attractive proposition than if we just include normal, your usual TB mortality and morbidity. We need to think about how the increase in mortality and morbidity in TB from post-TB could impact resource allocation across disease areas and perhaps also across geographies. We need to identify populations who are at greater risk of post-TB sequelae and think about how we make evidence which shows the burden, where the burden sits and how we can tackle it. And it's difficult to make convincing arguments for the financial support and social protection that is needed to support people living with post-TB, but we don't know what those solutions are, so we need to think about those areas. This slide summarises those kind of discussion points from the last two comments. I'll say thank you very much for listening.
Thank you very much. We are now going to move on to Gabriele, who is associate professor at UCL and clinical consultant at UCLH. His interest is very much about host-directed therapy, and he's going to speak to us about rational strategies for host-directed therapy to minimize post-TB lung damage.
Hello, everyone. Thank you for giving me the opportunity to talk to you today about strategies for use of host-directed therapy to minimize post-TB lung damage. This slide requires no introduction to this audience, as we all know that TB infection results in robust inflammatory response, histologically characterized by the granuloma, which can both support containment and protection from TB infection, but can simultaneously also drive pathology and disease through the immune cells that are recruited to that site. And indeed, it is this excessive inflammation that is thought to be responsible for a significant amount of lung defects in lung function following pulmonary TB, including distortion of airways resulting in pulmonary cavitation. So an important question is how significant all this is, what are the drivers, and whether any of it can be reversed. Unsurprisingly, the more extensive TB lung disease is, in terms of number of lobes involved, results in greater impairment of lung damage as quantified by FEV1 or forced expiratory volume. But even more strikingly, the reduction in FEV1 that is observed can persist even beyond the end of TB treatment. And that is important because reductions in FEV1 is associated with greater increase in overall mortality from all causes, demonstrating the significant burden that TB can cause even after the infection is treated. And so to summarize the problem that we have here, what we know and want to try and understand is what components of the inflammatory response after TB infection can support control of bacterial replication and resolution, in contrast to the ones that can drive tissue damage and post-TB lung damage. And that is specifically the role of host-directed therapies that try to dampen this pathological inflammatory response. Now, this concept is not new. Robert Koch described this many years ago, injecting homogenates of Mycobacterium into the skin of patients with TB disease, resulting in significant inflammation and necrosis of the injection site. And this has been recapitulated in mice, whereby injection of additional dead Mycobacteria enlarges the size of a granuloma compared to mice only receiving live bacteria. And crucially, in both settings, there was no improvement in control of bacterial replication. Studies have also explored the role of enzymes that degrade the extracellular matrix, such as matrix metalloproteinases, and these are elevated in the bronchial lavage of patients with TB disease, and these enzymes can result in destruction of the alveolar architecture and promote tissue destruction, cavitation, and erosion of bronchi and potentially transmission. So therefore, knowing that immunopathology is a feature of TB infection and TB disease, can we attenuate this? And actually a number of studies and candidate drugs have been looked at the moment in various phase studies, and these are a non-exhaustive list of the drugs being investigated. Last year, phase 2 randomized study was published looking at these drugs as adjuncts to antibiotic therapy in TB, looking at FEV1 as an outcome measure, and there was a hint that everolimus, the mTOR inhibitor, and the CC1050 phosphodiesterase inhibitor may have accelerated recovery in FEV1 faster than controls. But crucially, there was no signal for impairment of microbiological control supporting safety of this approach. Equally, last year, another study was published looking at doxycycline as an adjunct to TB drug therapy in pulmonary TB, and this demonstrated that indeed it resulted in impaired or reduction in MMP1 activity in the sputum, and though it's probably behind my video, there was a reduction in lung cavity in cavity volume in individuals who received doxycycline, hinting that there was a beneficial clinical effect. However, these sort of the kind of small changes observed in these two studies may reflect the sample size, but equally may reflect that the target was not correct. And knowing what is the right target for host-directed therapy is non-trivial. In part, that is because we cannot readily take serial lung biopsies to study the biology at the site of TB disease, but even if you could, you still have the problem of chicken and egg because of the confounding of disease, and you don't know what's cause or effect of what's being sampled. And finally, it's also important to mention that TB is a human disease, and so therefore a number of studies performed in animal models may not be able to fully recapitulate human pathophysiology. So we've tried to circumvent some of these challenges by repurposing the tuberculin skin test that everyone in this audience knows as an investigational tool, by biopsying the site of a TST and performing whole genome transcriptional profiling. And in so being, we create a model where we're measuring human in vivo tissue immune responses, and by having a challenge model, we'll be able to hopefully discern cause and effect by standardizing essentially the experiment. The site of a TST results in inflammatory responses that we all know and are familiar with in TB, and this inflammatory response actually shows strong correlation with variability in the responses that we observe between human TB granuloma and healthy lungs, indicating that the TST is a good model for affecting the pathology of TB disease. So we used the TST challenge model in a study that was published last year, looking at patients with active TB versus controls with latent TB, and found a whole group of genes here in red which were induced to a greater level following the same antigen stimulation sampled at the same time in these cohorts. Notably, one of the most prominent differentially expressed genes was MMP1, expressed at a much higher level in active TB. We were also able to try and predict the cytokines that were regulating this differential gene expression in active TB, and interleukin 17 or IL-17 popped out as a key coordinator. Now, why is that important? Well, we know that IL-17 regulates a large number of inflammatory responses, including tissue inflammation, chemotaxis for neutrophils, and induces MMPs. And most importantly, IL-17 is a known driver of disease and a number of chronic inflammatory conditions in humans, such as psoriasis and other causes of inflammatory arthritis. So we were able to go back and actually confirm that the IL-17 activity was indeed elevated by making functional cytokine activity assessments, and this was specific for IL-17 and not all cytokines such as interferon gamma. This was accompanied in active TB by increased enrichment of neutrophils in the TST of these patients, and also T cells producing IL-17, so-called Th17. And we supplemented these studies by performing the ex vivo stimulation of monocytes from these cells to see whether these cells had the potential to promote Th17 responses. And indeed, we saw that monocytes in active TB are able to induce high level of interleukin 1 beta and interleukin 6 cytokines, and so therefore these cells may be promoting further Th17 differentiation, which in turn are drivers for elevated IL-17 activity in TB. So putting all this together in our schematic of potential host-directed therapies, our work puts forward the idea that others previously not considered targets for host-directed therapies include interleukin 1 beta, 6 and 17, and these studies should be going forward to explore both mechanistic and clinical outcomes. But that's obviously not so straightforward, because the further hurdles are down the line, because how do you really make assessments of host-directed therapies? And I think this is more a general point that we should all consider. How do you measure the success of host-directed therapies? We've looked at clinical measures, so FEV1 or lung function, but also you can make assessments in radiological measures such as CT PET, and whether that's the optimal way of assessing the impact of host-directed therapies, microbiological assessments we've talked about. Perhaps we should also explore immunological assessments to try and quantify some of the dysregulated inflammatory responses we've seen and see how they diminish over time on TB treatment and whether that's accelerated by host-directed therapies. I think it's crucial that we also consider that this is not just happening at the experimental setting, and it involves patients, and so questionnaires to assess patients' experience and symptoms are crucial. And finally, timing is obviously important. When is the right time to intervene, in which patients, and when's the right time to assess? And some of these are still unanswered and open questions. But hopefully I've shown to you today that pulmonary TB results in significant lung damage, and that may be associated with pathological immune responses, but that understanding the mechanisms of the disease can inform the design of rational host-directed targets. Nevertheless, I think we still have a long way to go to try and understand and define the correlates of TB pathology in order to address heterogeneity between individuals, and also to guide our assessments of clinical outcomes in these patients. The work I presented today has been a huge amount of work from a large group of individuals, and I thank them, and I thank you all for listening, and I look forward to taking your questions in the Q&A session later. Thank you.
Thank you very much, Gabriele. Fascinating talk. And our last speaker in this session is Toyin. He is Associate Professor at LSHTM. He is also a clinician scientist at the MRC unit in the Gambia. He will talk about post-TB lung health in children, so a very specific group, but obviously a group which is, has a long life in front of them. So that is impacting potentially on a long period of post-TB lung disease in front of them in children in West Africa. Over to you, Toyin.
Good day. My name is Toyin Togun, and for this World TB Day Symposium, I will be presenting our work on post-TB lung health in children in West Africa. For disclosures, data that I will be presenting are from research projects funded by UKRI GCRF Award and the Wellcome Trust Institutional Strategic Support Fund. The opinions expressed in this presentation are solely those of the presenter and not those of the funder. Several studies have provided overwhelming evidence of relatively higher morbidity and mortality after apparently successful TB treatment, including reports of persistent lung function impairment. This slide is just showing a snapshot of some of the published studies. However, this evidence of a relatively higher morbidity and mortality after TB treatment in the published literature is almost exclusively derived from studies in adult population. Although pulmonary tuberculosis accounts for at least 80% of the estimated TB cases in children younger than 15 years of age, there are currently no published data about the long-term impact of pulmonary TB, including changes in lung function in children and adolescents who have completed TB treatment, especially in low and middle income countries. In this regard, over the past couple of years, we have successfully established a program of research and training on post-TB lung health in children within the Childhood TB Research Group at the MRC units in the Gambia. And we've built upon this work to extend our work to other countries in the West African sub-region. To highlight the work in the Gambia on this topic, I will highlight the work of my PhD student who is based in the Gambia, Dr. Essin Nkerewen, who is a pediatrician, and he has established the capacity for pediatric spirometry within our Childhood TB Research Group. For the first part of his PhD, he conducted a cross-sectional comparative study at the MRC units in the Gambia, and the flow chart to the right side of this slide shows the steps toward the recruitment of the participants. He enrolled 68 post-TB cases who were children younger than 15 years of age at the time of their TB diagnosis, and successfully completed their TB treatment not less than 6 months before the date of enrollment. And he also enrolled a comparison group of 91 age-matched children who live in the same household as the post-TB cases, but had never been previously diagnosed with pulmonary tuberculosis. He performed lung function measurement on all the children, and the spirometry data was standardized using the Global Lung Initiative 2012 African American reference range that have been previously validated among African children. This table shows the spirometry parameters of these children stratified by the TB status. You can see that the forced expiratory volume in 1 second, the forced vital capacity, as well as the FEV1 FVC ratio were all significantly lower in the post-TB cases relative to the comparison group, while the prevalence of abnormal spirometry in the post-TB cases was 38.5% compared to 17.4% among the comparison group. However, it is important to note that while the comparison group were children who've never been diagnosed or treated for tuberculosis, the prevalence of lung function impairments among them was found to be almost 20%. And in the analysis of the baseline characteristics of this study population, we found no significant differences in the distribution of exposure to environmental tobacco smoke or household biomass smoke exposure between the post-TB cases and the comparison group. This second table shows the multivariable analysis investigating the risk factors for lung function impairment in all the children. And they found that post-TB case as a proxy for previous TB treatment, as well as a self-reported history of frequent or repeated cough in the preceding 6 months were strong, significant, and independent risk factors for lung function impairment in the children. To our knowledge, this is the first study to assess lung function 6 months and beyond after TB treatment in African children, and this manuscript is just about to be submitted for publication. With regard to our work in West Africa, in 2020, we were awarded a UKRI GCRF award for a multidisciplinary project to begin advocacy and research towards increasing the awareness of post-TB lung health, specifically in children. And this project had both a qualitative research and a capacity building component. And with the work that we've carried out in this project, we've now established a platform for research on post-TB lung health in children in West Africa. With the capacity building component of this project, while COVID-19 caused significant delay, it also provided an opportunity to be innovative. And in this regard, I led the development of the first online course on post-TB lung health in children and on pediatric spirometry in collaboration with the Pan-African Thoracic Society. This online course took the foundational spirometry training of parts to the next level by incorporating multimedia components in English and in French. And with this, we were able to train 2 cohorts of healthcare workers comprising more than 50 healthcare workers from Anglophone and Francophone West African countries who completed the training, and they used it to start accumulating credits towards spirometry accreditation by the Pan-African Thoracic Society. We also used it to competitively identify participants who were then sponsored to an in-person practical training on pediatric spirometry and post-TB lung health in children at the MRC unit in the Gambia. This training took place in October 2021, and we had 20 healthcare workers from 6 West African countries who had the training, which included several theoretical sessions, as well as practical sessions, including calibration of the spirometer, as well as practical session on performing lung function measurement in children. These healthcare workers have now returned to their respective institutions where they have established capacity for pediatric lung function measurement, or they have contributed to strengthening already existing capacity for such. And we also use this training as an opportunity to provide a link between the healthcare workers in their respective country and the PATH spirometry manager for continuous support. With a fund from this award, we also bought and donated 2 brand new spirometers to 2 of the largest tertiary healthcare institutions in West Africa, including the Jos University Teaching Hospital in Jos, North Central Nigeria, as well as Kolebu Teaching Hospital in Accra, Ghana, specifically for dedicated pediatric spirometry capabilities at this institution. With this project, we have now established a multi-country and interdisciplinary network of pediatric lung health investigators across West Africa, and we're currently developing grant applications that will build upon this project to conduct a multi-country prospective study that will generate unique data from clinical, epidemiological, and applied health research into post-TB lung health in West African children. And to conclude, I would like to acknowledge colleagues in the Gambia, UK collaborators, local, regional, and international organizations that are partners, as well as the funders. Thank you very much.
Thank you very much. If all panelists could please unmute themselves, not unmute themselves, but at least they put their videos on. First of all, thank you again to Rahim for sharing this incredible story. Thank you, Hanif, for doing the interview and letting us really giving us a snapshot into what it means to have such a debilitating disease, but also that it doesn't stop at the end of treatment. And if I may just take the prerogative of the chair and ask Hanif, is there a place? Clearly, Rahim has said, it was her son who helped her to do little exercises to strengthen her muscles, to help her to actually get out of bed. She switched the telephone off because she didn't want to interact with anybody. Is there a place for actually more rehab, for physiotherapy, for the healthcare system to provide more support beyond just providing treatment and tablets, and especially also more support after TB treatment?
Thanks, Katharina, and again, just want to echo that. Thank you, Rahima, for spending the time to tell us about the story. It was a very powerful way to open this session and the whole meeting, I think. But I think, yes, you're right. There are several challenges to this. I mean, and the other thing that was quite striking about Rahima's story is that the delays in diagnosis, initially seeing physicians several times, and of course that's the flip side of this, is early detection is going to prevent many of these problems. The middle bit of host-directed therapy, I think, is important to research on, but we need to remember that actually at the end, what can we do for patients? How can we identify the patients that are having difficulties, and how can we best support them? And I think pulmonary rehab is definitely one of those things. And at the end of this treatment sort of was at the end of 2020, so right before the sort of peak of coronavirus, which kind of had it gives its own challenges about getting imaging and getting the right clinic appointments. So I definitely think there can be improvements in how we identify people that need ongoing support, and what we might be able to provide them with regard to rehab. I did think what was really striking about what you said was the impact on quality of life, and just echoing what Gabriele said in what should the outcome measures for interventions be. I think patient-centered outcome measures, quality of life outcome measures are clearly very important.
Thank you very much. Completely agree, and we have a couple of comments also in the Q&A saying that we haven't really put any focus, especially as national TB programs, who mainly focus on the people to be found and then to put on treatment, but we haven't really established any kind of long-term care following TB. Now there is a question specifically, I might direct that to you, Ryan, about the kind of standardized mortality ratios, and if some of these standardized mortality ratios actually have to do with like risk factors such as poverty, low malnutrition, diabetes, and if in these standardized mortality ratios, these factors are taken into account.
Thanks. Yeah, it's a very good question from Graham. The short answer is no. So the Romanowski paper takes into account general population, sometimes age and sex matched, but not matched for these factors, and we know determine a lot of the risk of TB, but also have consequences for general quality of life. But to say that, as Matt already raised, is that it's very difficult, which is part of this question, to get the causally ascribe the less the less high function, the lower lung function to the TB, or whether it's caused by the factors that led to the TB disease episode. What we did try to do in that paper is to try and isolate the causal path by looking just at the pulmonary pathway and look at the impact, the increased risk that the TB episode brought. Whether we did that precisely is impossible to know, but we did try to get there and be rather conservative. And just as to highlight Tom Yates' question, like that's just trying to get to the pulmonary consequence of TB that ignores still the impact on socioeconomic status, on stigma, mental health, etc., etc. So overall, the impact on general well-being in Delhi's is still strongly underestimated properly. But yeah, it is difficult to make that comparison very, very clearly, and more data is coming through.
Thank you very much. Marc, I hand over to you.
Thank you very much indeed. And I've got some questions for Gabriele's presentation. Question from Liz Corbett. She says, tuberculin skin testing in active TB, that's very interesting. What about the 20% or so of patients with TB disease who have no reaction to TST? Do they have less lung destruction too? Gabriele.
Thanks, Marc, and hi, Liz, and thank you for the question. It's a good question, and from my understanding was that those individuals who have TB disease but negative TSTs don't have worse lung damage, but that's worth double checking. I think it should also worth saying that in sort of a latent TB cohort, negative TSTs have been associated recently quite with insufficiency or inability to generate TB-specific interferon gamma responses, and that's not a cytokine that we saw a difference in, and we wouldn't, from our data, certainly anticipate to drive more lung damage. So I think the answer is probably not. We could double check that, but that's a good point, worth considering.
Thank you. And while you're being prodded a bit, here's a question from Willem Hannekom, who says, the dilemma with IL-17, which is what you were discussing, is that the responses also appear to be emerging correlates of protection against TB in animal vaccination studies. And there's similar suggestions in human clinical studies as well. So how do you answer that?
Yeah, it's a good question. Data? Yeah, thanks for prodding me, but thanks, Willem, for the question. And it's a good point, and actually this has been raised before. I think it's important to state that when looking at the literature of immune responses in TB, many cytokines and many pathways can have dual roles. I think everything is about context, and there are multiple examples where a particular pathway or cytokine can be protective in early stage disease, whereas and perhaps cause more tissue damage later. And I think that's where the literature together seems to be heading in terms of where the role of IL-17 is, where there's undoubtedly protection early on in infection, perhaps response to vaccination, but actually in an non-clearing infection, then the sort of persistent antigen perhaps can drive more IL-17 responses that drive tissue damage, where basically it's doing more harm than good by that point. I think ultimately, you know, this is the only way we can answer this question is by testing that experimentally. Perhaps in animal models, but maybe more in sort of directly into human experimental medicine studies. And I think that will hopefully give us the answer for IL-17 and many other cytokine pathways and whether they're tractable therapeutic targets.
Thank you. And one more question from Harsh Sharma. And this picks up a little bit on the model studies. So he mentions that in cell culture studies, type 2 pneumocytes as well as endothelium seem to be important and potent producers of cytokines, including IL-6. And certainly if you were to talk to the type 2 pneumocyte people and the endothelial people, they'd be very upset if you didn't include them in this model. So how do we do that? Is it all just much more complicated than we're trying to do at the moment, or what do we do, Gabriele?
Yeah. So I think that's a really good point about the fact that we can't be too reductionist about understanding disease biology. And that's where I think that's the benefit of doing the kind of studies we've been doing, taking skin biopsies, measuring responses in whole tissues that include stromal cells like epithelial cells. They're not just particular immune cells you can isolate in culture. I mean, if you want to go down the route of trying to identify what cell makes what, I think the sort of single cell approach from a really mechanistic perspective is right. And then that hopefully will inform as to whether particular cytokine pathways like IL-6 indeed are overexpressed in TB and may well be therapeutically tractable, and then ultimately see whether they're beneficial to the patient. So I think it's a valid point. It's all about thinking about your model and how it relates to disease biology.
and not leaving any cell out. Don't want to be discriminatory.
Thank you. And now I'm going to ask one more question and then hand back to Katharina. And it's a question actually that no one sent in from the audience, but I was very interested in Toyin's presentation because amazing work and so important because completely neglected population. One of the things that I've discovered from some of the work and discussions I've had with people such as Kevin Mortimer is not only are there very few spirometers, but there's almost no lung function testing equipment in many parts of the country. Yeah. There's very few people trained to do it, and there's hardly any pulmonary rehab services available. So how do we approach that? And are there plans to do this? Because it's something that here based in London, no one would even think about until you actually look at the problem.
Yeah, I think the key thing is to start that's why advocacy is really very important. Raising awareness about the importance of post-TB lung health, which is one of the reasons why there might be need for spirometry. I mean, there are many other reasons, asthma or the COPD and things like that. But specifically with regard to the measurement of lung function test following TB treatment for tuberculosis, it's important to actually improve the capacity and the confidence of healthcare workers to be able to perform lung function measurement. And that's one of the things that we've been, from the project that I've been leading in West Africa over the past couple of years, we've been trying to do that across the region by training not only physicians, but actually senior nurses, who were the ones that could actually often be frontline health workers. And we from the fund, from the grant, we also bought spirometers for some of the institutions, following the fact that we have established capacity for pediatric lung function measurement at the MRC unit in Gambia. With the program of research and training that we have now established the unit in the Gambia as a hub for research and for training sub-regionally in West Africa. And we're building upon that by extending the work. So part of the thing that needs to be done is not only engaging with the healthcare workers, training healthcare workers, but also engaging with policymakers. And we've started doing that early. It's important for policymakers to actually, for them to integrate this into policy, they need to be made aware from our interaction with policymakers. Most of them are not even aware of post-TB lung health being an issue. Some raised issues about what actually is the definition, which is genuine. They don't, what's actually is the definition of post-TB lung disease or post-TB lung health, which is actually a scientific question that we don't have clarity about. And what proportion of TB patients are likely to develop, persistent health impairment. All this information are needed for healthcare workers to fathom, resources allocation to the aspect of, post-TB lung health. So there's a need for more research. There's need for, clinical, epidemiological, and applied health research, qualitative studies that will generate sufficient data on post-TB lung disease across the life course, but importantly in children who bear particularly huge burden of tuberculosis, with at least 80% of TB disease in children being pulmonary tuberculosis. Yeah.
Thank you, Katharina.
Thank you very much. So Toyin, I would have lots of questions for you, but I will stick to the questions in the Q&A box. I just wanted to also highlight Marc's comment and the funding link for the National TB program in Kiev. But then there are two comments both from Jayne Pillai and from Les Corbett about, how can we move the agenda forward with regards to, I know you've said advocacy, with regards to improving the outcomes beyond just treatment. Yeah. So Liz asked if there is any, if anybody has experience with, for example, post-TB clubs, or if there is something which could be done very practically. I mean, I know that respiratory physiotherapy, even in the context of COVID, is just almost non-existent. But if there is actually almost like training of nurses by physiotherapists, which would be potentially able to actually administer, or even lay healthcare workers, which would be able to administer something along the line of a pulmonary rehab. Over to you, Toyin.
Yeah, that is exactly one of the things that we have sort of like established within collaborative partnerships in West Africa from the work that we've been doing. And the unique thing essentially is that we importantly made this engagement with healthcare workers as well as policymakers, and importantly, civil society groups, as well as TB advocacy groups across West Africa, working in collaboration with TB Proof in South Africa to ensure that we have input from, you know, the TB patients who have actually completed TB treatment, from TB survivors, basically to have a good understanding of, you know, how they handle area of persistent health impairment, you know, following apparently successful TB treatment, and providing the sort of like interaction between the healthcare workers, policymakers, and TB survivors is a good opportunity to actually start the process of advocacy. And building upon that with training, we have now established a network of, you know, pediatric lung health investigators, as I mentioned, across West Africa, with linkage to the Pan-African Thoracic Society's Spirometry Manager. And a refresher training is actually, and we have WhatsApp group for all the people that we've trained, nurses and doctors who are linked to the Spirometry Manager. And there's going to be a refresher training in a couple of weeks, sort of like all of them are working towards submitting their protocols, updating them. This is the way to actually start, you know, building confidence and capacity of healthcare workers to perform lung function measurement. With that, we can start generating data, getting better understanding of actually how many people develop health impairment, lung function impairment following TB treatment. That is information that policymakers will need to know for them to integrate this into their policy agenda. So it has to be an integrated work with working between researchers, policymakers, and the civil society organizations, as well as TB survivors, which is a platform that we have now established within West Africa.
Thank you very much, Toyin. If I may just say, we also need to think about more than just the lung, because especially for the adolescent, they are in their growth spurt and they might never actually get to their full potential with their skeleton, then leaving them quite vulnerable to fractures later on, as we have seen with a lot of adolescents with HIV. So I think specifically because TB interacts also with vitamin D, I think we really need to think beyond the lung. I hand over to Marc. It was an absolute pleasure to chair this session. I have learned tremendously. Thank you very much. And I'm excited to hear all of the other talks which are going to follow. Thank you.
Thank you, Katharina. Thank you, speakers. Thank you for putting the videos together. Thank you, of course, to Rahima for a fabulous presentation. And now I'd like to move on to the next session and welcome the hosts for Understanding M. tuberculosis in the Post-genomic Area, handing over to Isabel and Sam. Thank you.
Thanks very much, Marc. We're now going to move on to the next session, as Marc said, and we're going to think about some available technologies and how they might provide new opportunities for those with TB into the future. Now, I'm very pleased to introduce our first speaker of this session I've worked with for many years, and that's Professor Tim McHugh. Tim's Professor of Medical Microbiology and Director of the UCL Centre for Clinical Microbiology, and his primary research focuses on infections of the respiratory tract, with particular emphasis on drug development for TB. Tim is going to speak directly to our theme in this session with his talk entitled Post-genomic TB Research and its Role in Diagnostics and Drugs. Thank you, Tim.
I'm Tim McHugh, and I'm going to talk about Mycobacterium tuberculosis diagnosis and treatment in the post-genomic period. And so what do we mean by the post-genomic era? Well, the first thing is to think about the whole genome sequence of MTB, which was first published in 1998. That was a step change in our understanding of Mycobacterium tuberculosis, and also the tools that we could start to apply to understand the biology of this organism. Of course, the other thing which is a major step change in our ability to explore the biology of all organisms was the introduction of next generation sequencing, and the ability to sequence whole genomes of bacteria in a timely way, but also at a cost-effective manner, has enabled us to move very quickly to understand the detail of the biology of these organisms. And an example of the importance of that is shown in this cartoon from Giovanni Satto's PhD, published in MRC Medicine. Whereas that first image I showed you was H37RV TB, one genome representing several years worth of work and lots of money and lots of people's effort. This is the genome of a dozen or so strains of Mycobacterium tuberculosis from an outbreak. And Giovanni was able to align these and explore the transmission routes between these strains in the context of a single PhD. So what do we need to understand about TB in the post-genomic era? When I was thinking about this talk, I was trying to think what would capture all those elements. In fact, I gave up and asked my colleagues in the Centre for Clinical Microbiology to help me out. And so they created this word cloud for me. The first thing which we focus on is the resistance phenotype versus its genotype. So we now have the ability to describe drug resistance patterns in terms of the genome of the organisms, and that's something we'll look at a bit more in this talk. But of course, the genetics of resistance, whether that's the evolution of resistance and the epidemiology of resistance, or whether it's the understanding the mechanisms of drug resistance, well, then that's becomes really important, and that was highlighted by Marc. We've spent some time exploring the physiological states of Mycobacterium tuberculosis in the context of its action, or the action of different drugs at different stages of the life cycle of the organism. And that came through as something that we need to highlight in this talk today. And then, of course, the final bit is the real business that we're interested in, clinical applications, whether that's drugs or treatment. And the post-genomic era has given us major insights in that area. So the first thing to think about is the resistance phenotype versus its genotype. This is a conventional phenotypic study for drug resistance. I have to admit this is Mycobacterium abscessus, not MTB. And Priya, who's a PhD student and lab manager in our group, has been looking here at bedaquiline and clofazimine and then together at the MIC, or the drug susceptibility pattern of these organisms in this conventional system, which involves growth of the organism in appropriate media. However, we can expand that by using genomic methods, and we can look at not only the phenotypic resistance to resistance, but we can align the genome and look for the mutations which encode resistance. And this work, led by Amanda Brown, does just that, using clinical isolates and comparing the culture phenotype with the genotype that was obtained from them. And although in most cases, here you can see pinkness, you can see that the things are aligned. But in this case, and in these cases, there is a distinct difference between the phenotype and the genotype. And understanding, though, not only understanding the mechanism of that, but also the importance of that is important in how we interpret our genotypic data. That's been expanded or emphasised in this piece of work, which Anna Bateson has just published in JAC. And to cut a complicated story down, this is looking at protominid resistance or susceptibility to protominid by a range of clinical isolates. It's a surveillance study in which we had help of 5 or so laboratories around the world, and we just screened a whole range of strains to find what the cut-off points should be for the difference between resistance, which is this red spot here, and susceptibility. Now, when we did the phenotypic study, we found this biphasic pattern for Mycobacterium tuberculosis with 2 peaks and 2 potential cutoffs. How would we unpick that? Well, we unpick that using genomic data. And if you look at this cartoon here, you can see that the bright green is all around here. This is all the same strains, and the dark green is all around here. And when we look at the genotype of those dark strains, we find that they're all members of the L1 lineage. That's the Indo-Asian lineage. So we're seeing a distinction in drug resistance pattern between most lineages of TB and the Indo-Asian lineage, or L1. And that becomes important not only in designing our diagnostic tests, but it also begs the question about how we use protominid in that population, and whether different drug levels are needed and how we interpret that data is needed. But of course, the genetics of resistance is not only about tracking antimicrobial resistance. We can use the genome to explore the mechanisms of activity. And this is illustrated by this work, again from Giovanni's PhD, in which he looked at PAS, para-aminosalicylic acid, and we were looking for the mechanism of action of PAS. So he designed he developed some resistant mutants and then probed the genome by doing whole genome sequencing to look for differences between the resistant strains and the sensitive strains. And there's just 2 levels of drug here used for selecting the mutants. And we found that, yes, there were specific mutations in specific genes which help us understand the mechanism of action. And particularly in this case, FOLC, which is a folate metabolism gene, was found to be specifically associated with resistance and had not been described previously. So it's a new variant associated with resistance. The other thing about the genetics of resistance is we can start to explore the evolution of resistance. So I've already shown you this picture from Giovanni's work, looking at the evolution of resistance in an isonized resistant strain moving around North London. But by aligning the sequences and looking for subpopulations from a single isolate, we can start to see subpopulations within that isolate and start to explore the evolution of resistance within that isolate. And as we said, as myself and Gwen Knight and our PhD student Jayne Fonseca said some while ago now, the mechanisms and pathways that result in the emergence and subsequent fixation of resistant strains are not yet fully understood. And in fact, the work I'm doing with Gwen and our new PhD student, Naomi Fuller, is to use mathematical modeling to try and take this data and parameterize it with some biology, but also predict the outcome of subpopulations of drug resistance strain isolates within a single isolate. So we're starting to try and map that evolution. Of course, once you start mapping evolution, you want to start thinking of experimental models to test that. And here's an example of one. This is the hollow fiber infection model, which is basically a way of culturing organisms and then feeding them nutrients or drug in a systematic way so that we can look at, in a dynamic way, at the evaluation of the drug-bug interaction. And Frank Lefogh, who's a senior fellow at UCL, has done this work in TB, and we did this work looking at the evolution of isoniazid and rifampicin resistance in this hollow fiber model over time with different levels of drug. And you can see that there's 2 cycles of drug, and what he's done here is map the genotype of the resistant strains as the experiment goes on. And from here we can see the impact of the drug on the evolution of resistance, and that is defined by the mutations that we're observing. So we can see the emergence and fixing of resistant genotypes in the whole fiber model. Thinking about physiological states, moving away from that genotype to the physiological status of the organisms, well, we know that TB is in different physiological states. And one of the pieces of work that we've done, in fact, Isabel, who's chairing this session, did this work using a molecular tool to explore the different populations of cells within a culture. And so she developed the molecular bacteriological load assay, which measures 16S rDNA. And it's a quantitative amplification which enables us to quantify the numbers of bugs in a particular culture. What was important here is that the number of bugs seen here is different to the conventional counting method on solid AML. And what we see here is not an artifact of the methodology. What it is a reflection of the method of measuring, detecting a subpopulation of cells which are not seen on solid media or indeed in liquid media. So that approach has been expanded by Dimitrios Evangelopoulos, who's going to speak later. And here he used those same methodologies to demonstrate the fact that the MBL assay is picking up different populations in this experimental mouse model of drug treatment over time. And so you can see that the conventional culture methods were not picking up the same numbers of cells as the MBL and the MPM method, and this reflects that different populations being detected. But we need to think about clinical applications, and this is the final section that the group identified. So molecular tools have enhanced our ability to detect Mycobacterium tuberculosis, and also to define the drug-resistant pattern. The Cepheid gene expert has been a game changer in this area, mandated by WHO in 2011 and rolled out across the world. It's a PCR-based methodology which detects TB DNA. Now that's great, and it's been a major success. We feel that we can expand on that, and so using the MBL assay that I've just described for measuring cell numbers, we've been able to use this, and we're beginning to use this as a tool for monitoring therapy. So because it measures viability rather than just DNA, we can use it for monitoring. We're using it for monitoring therapy. And drug resistance, well, the gene expert gives a limited amount of drug resistance, but genotypic methods enable us to explore those in greater detail. And this is just an example of the diagnostic test, the Hain assay, which detects drug resistance across a whole range of genes. Another clinical application, which is perhaps in the future, is using our combination of conventional microbiology and molecular methodologies to optimize mycobacterial treatment. And this shows synergy energy testing with by growth of organisms, transcriptomic profiling of the genes expressed during drug treatment, and then seeking associations between drugs to help us identify the optimal drug treatments in an individual patient or in a clinical trial in the best regime to take forward. The final area that we need to think about is epidemiology, and I admit this isn't an outbreak dendrogram. It's actually a case looking at patients in a drug trial and looking for relapse versus reinfection. But just to illustrate here that you can, using the whole genome sequences, you can align strains and look for patterns of transmission and associations between strains, which has again been a step change in our understanding of the distribution or dispersion of TB in communities. So understanding Mycobacterium tuberculosis in the post-genomic era. We no longer are limited by the tools available to us. Well, we are a bit, but it's fantastic. Conventional microbiology is enhanced by genomic analysis, and it's important that we do these two things together. It's driving discovery science right at the beginning, identifying drug targets, understanding the metabolism of organisms. It's translational science, enabling us to develop new treatments and new diagnostic tools. And actually, it's already, genomic methods have already moved into clinical practice. We're finding answers to long-standing questions. And importantly, these tools are enabling us to ask and explore completely new ideas. So we are at the beginning of our understanding of Mycobacterium tuberculosis biology, and it remains an exciting and fascinating challenge. Thank you.
Thank you very much for that interesting talk, Tim. I'll hand over to my co-chair now, Sam.
Sorry, my apologies. I have a technical issue.
Sorry, I think Sam's struggling there. So let me introduce our next talk. So I'm very pleased to introduce Dr. Gwen Knight. Gwen's from the London School, and she's an associate professor and co-director of the AMR Centre in infectious disease epidemiology. Gwen uses mathematical modelling to explore questions around the dynamics of antibiotic resistance. So I'd like to hand over to Gwen now. Thank you very much.
Hi, my name is Gwen Knight, and I'm an Associate Professor and Co-Director of the AMR Centre at the London School of Hygiene and Tropical Medicine. Today I'm going to present to you how mathematical models can be used to understand emergence of drug resistance in tuberculosis. Broadly, what we're interested in is this question here: who carries multi-drug resistant Mycobacterium tuberculosis? And in this presentation, what I'm going to talk about are two projects, one which focused on those carrying resistant Mycobacterium tuberculosis as a latent infection, and those that had symptomatic disease. So first of all, I focus on those with a latent infection, and we wanted to know who carries multi-drug resistant Mycobacterium tuberculosis in this asymptomatic state. And to do this, we took estimates of the annual risk of infection, or the risk of infection with Mycobacterium tuberculosis, from the 2016 estimates of the global LTBI burden. And we combined these with WHO data on the proportion of infections that were with multi-drug resistant, or MDR, Mycobacterium tuberculosis. And this data came from both surveillance and surveys. We assumed that rifampicin and isoniazid-based TB treatment was introduced in the early 1970s. So combining this risk of infection with the proportion of infections that are with multi-drug resistant Mycobacterium tuberculosis to an age structure cohort model, what you can do is you can track over time an individual's risk that they get infected with a resistant or susceptible strain. You can do this over a population to try and work out the prevalence of infection with resistant or susceptible Mycobacterium tuberculosis. When we did this, we made several assumptions, the two main ones being that we assumed lifelong infection and an 80% protection from reinfection. What we found was substantial global heterogeneity. We estimated that on average, the global prevalence was 0.3%, with a 95% uncertainty interval between 0.2 and 0.3 in 2014. We estimated that multi-drug resistant strains accounted for about 1.2% of all latent TBI infection. And you can see the spatial heterogeneity in the map here. What we also found was substantial heterogeneity in the burden by age. So you can see here the trends, the WHO regions. So on average, 3 in every 1,000 people globally were estimated to carry latent MDR TB infection. We estimated that prevalence was around twice as high in those younger than 15 years. So this project here demonstrates that we can use mathematical modelling to combine different pieces of information to estimate something that's highly important for understanding who should receive prophylactic therapy and who should also, who is at greater risk of progression to TB disease with a resistant strain. However, this is something that we can estimate only using mathematical models, as it's very hard to know exactly what type of strain or what the characteristics of the strain that are causing latent infection. So the second project I want to talk about focuses on those with active disease. So who has multi-drug resistant tuberculosis? When we say that someone has multi-drug resistant tuberculosis, what do we really mean? So we've got someone with symptoms and we take a clinical sample from them. And then, depending on the context and setting, we determine that this sample, the bacteria within this individual, have antibiotic resistance. And we either do that genotypically or phenotypically. But at every point in this, there is a bottleneck. We can't sample all the bacteria in the patient, and we also don't usually get quantity, or we don't quantify the diversity in that individual. We're interested in the presence or absence of of strains that are resistant. And phenotypically, what we mean is that we could detect bacteria within that patient that grew in the presence of antibiotics above a certain concentration, above this pharmacological breakpoint. But what about all those bacteria that are potentially in the patient that would grow in slightly raised levels of MIC, of antibiotics? So they don't have a totally susceptible phenotype, but in fact, they survive in low levels of antibiotic. Are they important? Do they compete with the resistant strains? Are they linked to clinical endpoints, for example, treatment failure? It's something that we don't know because we don't explore them. The tools that we use, we're looking at this threshold. We're only interested in the presence or absence of Mycobacterium from that individual that are highly resistant. Now the question comes whether this matters, because in terms of antibiotic therapy, you just want to know whether your therapy is going to work. Now, if we think about a different bacteria, so methicillin-resistant Staphylococcus aureus, some work that I've done here has looked at how many samples we need to take from a patient, or how many single colonies we need to look at in order to understand the distribution of resistance. In effect, what we did was we looked at individual patients, and we looked at single colonies from those patients and looked at the individual antibiograms that those single colonies had, so those individual bacteria. And then we used modelling to infer how many samples you'd need to take on average to have to see all resistances that exist within that patient's Staph aureus population. And we found that you'd need something like more than 25 individual colonies to get a greater than 95% probability of seeing all resistances. So all combinations of resistances, but also all resistances within that patient. And so that's obviously important because if you do sample single colonies and just test one of those for its resistance profile, then there's a high chance that you will that you will miss many of the resistances, or a substantial proportion of the resistances in a Staph aureus, in a patient Staph aureus population. I think this matters for Mycobacterium tuberculosis because we're now seeing a greater range of treatments and a greater range of antibiotics that we can use to treat Mycobacterium tuberculosis, and we're starting to enter an era where we need to be thinking about different combinations, and to think how they might affect, or how those combination therapies, or how that diversity in the patient might be linked to clinical outcomes, and therefore how we can use treatment better. We already have signals that that there are differences in the mutations and the genetic profiles that can lead to resistance. So, for example, genome-wide association study recently from a large collection of strains in Peru found certain mutations that led to an increased risk of drug resistance acquisition. And so this diversity in the genomes, this selection for potentially for mutants that have some kind of stepping stone that allows for greater resistance later on. And what we're hypothesizing in Naomi Fuller's LIDO PhD is that there are this subpopulation of strains that have a decreased susceptibility, so are only partially susceptible, potentially could act as a stepping stone to full resistance, or in fact they might have an increased fitness. Some initial results suggest that we can find mutations like that, and so they might actually act as competition. And so what you might do in a patient is screen for these partially susceptible mutations to see whether they're more at risk for developing resistance within that patient, or whether they might act differently in terms of their resistance development risk. And so we're exploring dynamics such as those shown on the right-hand side, where the x-axis is time and the y-axis is the proportion of the population that consists of these different susceptible, partially susceptible, and resistant subpopulations. And when we started Naomi's PhD, what we were interested in is whether other individuals, specifically in the Mycobacterium field, had considered resistance heterogeneity and the appearance of mutations that may confer a slightly raised MIC, or this partial susceptibility. So to do that, we had to explore the total landscape of mathematical models that had looked at drug resistance in Mycobacterium tuberculosis populations, and what we found that there were approximately 180 of these, 184 in these preliminary results. Most of them looked at transmission models that explored interventions, the cost effectiveness, and most of them considered multi-drug resistant TB, so isoniazid and vancomycin resistance combined. Of those that specifically modelled one country, the countries most represented were South Africa and India. And these are preliminary results and something that we are currently working on. So in conclusion, I hope I've shown you that mathematical modelling can enhance data collection to estimate burden as well as explore evolutionary pathways. We found that MDR-TBI prevalence is around 3 in every 1,000, with a peak in younger ages. And we're now exploring this to see how much this influences disease prevalence, and we're looking at every AMR prevalence in other bug-drug combinations and how they vary with age. Mathematical models of resistant Mycobacterium populations have mostly focused on Mycobacterium tuberculosis to explore the impact of interventions with the transmission model structure for multi-drug resistant tuberculosis. An active question, and something that I don't think we know yet, is what is the contribution of partially susceptible pre-resistance mutations to resistance evolution? So watch this space. We hope to present something in future World TB Days about the importance of these mutants. I'd like to say thanks to Naomi, to Pete, to Finn, to Ryan, to Martin Chataka, and also to Tim. Thank you for listening, and I'll be available to ask and to answer any questions you have later on today. Thank you.
Thank you, Gwen. Our next speaker is Dimitrios Evangelopoulos. Dimitrios is a lecturer in microbiology in the Department of Microbial Diseases at UCL. He's based in the Royal Free in North London. And his lab is interested in studying the molecular and cellular mechanisms of drug action and drug resistance in MTB, with a particular emphasis on identification, verification, and development of novel anti-tubercular therapies. He'll be talking to us this afternoon on what drives resistance evasion in TB and can we learn lessons from cycloserine.
Hello everybody, thank you for your invitation to present our work in this session. I'm Dimitrios Evangelopoulos and I'm talking to you today from the UCL Royal Free Campus in North London, UK. Today, I'll show you findings from our research in cyclosporine resistance in tuberculosis and if we can delay drug resistance. Antibiotic resistance is one of the biggest health public challenges of our time. Infections that once could be treated can now be fatal. And more than 10 million of people could die of AMR in 2050. In addition, more recently, drug-resistant strains of tuberculosis have emerged and spread, making drug-resistant tuberculosis one of the major airborne drug-resistant epidemics today. It is harder and more costly to treat than susceptible TB and affects more than 500,000 of people around the globe annually. And so it is clear that new therapeutic interventions are urgently required for treating tuberculosis and managing the spread of drug-resistant tuberculosis. With a limited number of new antitubercular drugs being developed, it is important to identify ways to maximize the lifetime of the existing drugs, to save our drugs. Through our research, we're trying to solve these two questions. Can we delay the emergence of drug resistance? And what can we learn from antibiotics that evaded resistance the longest? Antimicrobial resistance quickly forms after the introduction of a new drug into the clinic, as you can see in this schematic here. There are only 3 antibiotics that delay the emergence of drug resistance the longest. These are glycopeptides such as vancomycin from the mid-50s to the mid-80s, polymyxins such as colistin from the mid-50s again to the mid-2010s, and in the TB field specifically, cyclosporine from the early 60s to the mid-2010s. This cyclosporine first discovered in 1955, and it's part of the second-line drug that we use to treat drug-resistant cases of tuberculosis. Mutations that are associated with cyclosporine resistance are not very well established in the clinic. We know that mutations in sequelae can confirm cyclosporine resistance in Mycobacterium bovis BCG, the vaccine strains of TB, and there were also reports about the involvement of alanine dehydrogenase and alanine racemase with conferring resistance to DCS in the clinic. As you can see here, DCS or dicyclosporine, it's a structural analog of aniline. Before we go into resistance, let's have a quick look and see how DCS works. DCS works by inhibiting 2 enzymes, alanine racemase and the diala-diala ligase. These 2 enzymes form the dipeptide diala-diala alanine that is an important structural part of bacterial peptidoglycan. In Mycobacterium tuberculosis, using isotope metabolomics, stable isotope metabolomics, it has been found out that actually the lethal target of DCS is the second enzyme, diala-diala ligase, and this is what causes the bacteria to die. In addition, more recent work from Lichara and colleagues also unexpectedly showed that alanine racemase inhibition by DCS is actually not permanent, and a small fraction of the enzyme is being reactivated via hydrolysis of DCS, adding further evidence and importance of the real target of DCS in Mycobacterium tuberculosis, diala-diala ligase. Now, when we're trying to look into resistance of drugs in tuberculosis, quite common we could see non-synonymous SNPs from clinical isolates. In this case here, you see moxifloxacin and rifampicin drugs and their respective targets, JRA and RPOB. You can clearly distinguish areas of hotspots of mutations were accumulated, especially non-synonymous mutations. When we look at the same for targets related to DCS, you could see that actually it's a very low abundance of SNPs, and that would really pinpoint to any involvement in mutations and resistant conferring mutations. We then wanted to check the mutation rate and calculate the mutation rate of DCS resistance. What you can see here is the mutation rate of isoniazid being 1 in 10⁻⁶ per bacterium per genome. and the mutation rate of rifampicin being somewhere to 1 to the 10 to the minus 9 per bacterium per genome, and the mutation of the rate of ethambutol and streptomycin somewhere in between the two. When we calculate that for cycloserine, what we find here is that this cycloserine has an extremely low mutation rate in Mycobacteria, to 10 to the power to minus 11. That in itself could explain why we don't see a lot of clinical resistance of this drug in the clinic. Through this experiment, the L'Oreal-Dorbert fluctuation analysis, we're able to isolate 11 mutants that were all resistant to DCS. We whole genome these mutants, and we find surprisingly mutations in alanine racemase, either mutations at that creating an aspartate to an asparagine substitution at codon 222 amino acid of the protein, or mutations upstream of the promoter or the specific repressor area. In addition, we also identify secondary mutations in some of these strains. One of the first questions we ask is actually, is there any cross-resistance to any other drugs? And we find actually that all of these resistant mutants were specifically resistant and 8-fold higher in MIC compared to wild type against DCS, but there was no other cross-resistance among first-line or second-line drugs. We then wanted to see if there is any fitness cause associated with these mutations. We did that using traditional competition assays in vitro, but also as we were researching Mycobacterium tuberculosis, we wanted to try in something more physiological relevant inside human macrophages, but also in an animal model of a mouse infection. This heat map here summarizes our findings from the fitness experiments, and what you can see is that actually some of the mutants have severe fitness cost, especially as it's more clear within macrophages and in the animal model of infection. And these are mutants 4, 5, and 6 in particular. And that, and again, could be actually explained why this severe fitness cost can actually stop the spreading of these mutants in the normal conditions. Then we want to see actually what is the mechanism of action of resistance against cycloserine and how these mutants actually became resistant. From our targeted transcriptomic experiment, when we checked the expression of different genes, we could identify a clear pattern. Mutations that were either at the promoter region or at the repressor region upstream of alanine racemase gene had an effect of overexpressing the gene on normal conditions as well as under cycloserine pressure, but also resulting in overexpression of a protein, in this case alanine racemase, as we could find out from the Western blot in mutant 5, 8, and 11. This overexpression of alanine racemase actually causes DCS resistance by having multiple extra targets of protein, so it can absorb the pressure of DCS. In addition, when we look at what happened on the aspartate asparagine substitution mutant at the position 322, we could see that actually this mutant enzyme is actually less inhibited by DCS by 240-fold less reduced inhibition activity, meaning that again it can actually be less inhibited and perform part of the job. But now, what are these findings tell us about the clinical implication of this work? What you can see here is the alanine racemase gene, and highlighted are various mutations that have been identified. With red here are mutations that have been confirmed to cause disease resistance, including our mutations here, as well as mutations upstream in the promoter region. And what we can do by having this knowledge is actually develop a molecular diagnostic tool so that we can screen and identify and detect disease resistance in patients early enough so that you can then have an appropriate treatment. In addition, these mutants are currently being aided towards optimization of drug susceptibility tests that you can perform to new clinical isolates to then establish if they're resistant or not against cyclosporine, as the protocol for DCS is not very well calibrated. And in addition, these methods could also be helpful towards evaluating of critical concentrations and cutoffs of DCS in the clinic. And further, of course, this highlights the importance of this pathway as well as the importance of having a molecule that has an extremely low mutation rate and further optimization of this derivative that keep these characteristics could be helpful for future drug discovery projects. So the take-home message here is that lack of clinical resistance to DCS in tuberculosis is most likely because of the ultra-low mutation rate, as no-cause genotypes, phenotypes can actually appear and propagate and spread during infection. And then, is drug resistance possible to evade? That's a big question, of course. And sadly, or what we can find out so far is that you can actually delay resistance and the evolution of resistance in the clinic, but it's always a matter of time. So it's a time game until resistance is established and being spread. This work was part of my postdoc at the Creek and was funded by the Creek at the Laboratory of Mycobacterium Metabolism and by the Research through supervision of Liz Carvalho, and worked closely with Max Gutierrez for macrophage assays. Teresa Cortez did the bioinformatics of the whole genome sequence. People at the National Institute of Biological Standards and Control helped with part of the animal experiments, and of course the amazing quick support from the STPs and the media. And thank you very much, and I'll be happy to take any questions.
Thank you, Dimitrios, for that really insightful talk. Our next speaker for this session is Dr. Terry Kipkorir. He is an Academy of Medical Sciences Newton International Fellow based in the lab of Dr. Christine Arnvig at the Institute of Structural and Molecular Biology at UCL. He obtained his PhD from the University of Cape Town under the supervision of Professors Valerie Mizrahi and Digby Warner, where he explored the production, acquisition, and utilization of coenzyme B12 in Mycobacteria. His current research is focused on understanding the role of vitamin B12 in gene expression in MTB. He'll be talking this afternoon on riboswitches, sensing and adapting to host environment via RNA through co- and post-transcriptional regulation. Thank you.
Welcome to my short talk on sensing and adapting to host environment via RNA through post-transcriptional regulation. My name is Terry from the Institute of Structural and Molecular Biology at UCL. We know that MTB can survive for many years within changing host environments which have different sets of stress conditions. And we think this is possible for MTB because it may sense and adapt to these changes by altering gene expression at the transcription or post-transcriptional level. And leader sequences of genes, which are mRNA stretches preceding coding sequences, are particularly a hub for post-transcriptional regulation through RNA binding proteins or RNA structures that can sense and bind small molecules and metabolites, or as binding sites for regulatory small RNAs. Our group has been interested in understanding regulation via RNA leaders in MTB to uncover those that may be associated with drug resistance. My colleague Alex has used next-generation sequencing. He has found that about 400 genes in MTB may utilize RNA leaders for gene regulation, and some metabolic pathways are particularly enriched for this type of regulation, and these include cell wall and cell processes, information pathways, regulatory proteins, and PEPPEs. And we have begun to look at, or our group is currently looking at, characterizing some of these leaders. Today, I will speak to you only about one class of RNA leaders known as riboswitches, which are stretches of mRNA which have two functional domains. These are regulatory segments of mRNA which have two functional domains. One is an aptamer domain which can bind a ligand, and the other is an expression platform which executes gene expression control upon a conformational change induced by the ligand. And this conformational change can either turn on or off the expression of the protein encoded by the mRNA. My project specifically looks at vitamin B12 riboswitches. Vitamin B12 is a cofactor required in all domains of life, but it is produced only in a subset of bacteria and in some archaea. And MTB does not itself produce vitamin B12. And so what I think is B12 is a host-derived signal for gene regulation in MTB. There are two vitamin B12 sensing riboswitches in MTB which are currently known. One occurs in the leader of the MET-E gene, and the other occurs in the leader of the PP2 gene, which shares an operon with B12 biosynthesis genes COBQ and COBU. To check whether these riboswitches are indeed functional, I generated a translation fusion reporter system by fusing MET-E or PP2 leaders with a LAGZ reporter, and put this in a plasmid where they have a strong MTB promoter, and put this in Microbacterium smegmatis, which produces vitamin B12, or in a mutant which does not produce B12. And what you can see here is that at the protein level, there is an attenuated expression of MET-E or PP2 in the presence of B12 compared to when there is no B12 seen here in the gray bars. And what I think is happening here is that these riboswitches are binding B12 and acting as off switches at the protein level. Now, what is happening at the RNA level? To check this, I incubated MTB cultures with B12 for 1 and 3 hours, and then I checked the transcription profile using North and blot analysis. And what you see immediately is that addition of B12 results in termination of transcription, or induces termination of transcription in the MET-E leader, but not in the PP2 leader. And what it means to me is that there are differences in the modes of regulation by the two switches at the RNA level, but which lead to similar outcomes at the protein level. And this result underscores the significance of looking at RNA leaders as hubs of gene expression regulation. Now let me speak a little bit more specifically about the MET-E switch. The MET-E switch contains 3 elements that are essential or known to be essential for riboswitch regulation. And one of those is the Shan-Delgado sequence, which is a binding site for ribosomes that are initiating translation. And the other component is called the anti-Shan-Delgado sequence. It is a sequester of the Shan-Delgado sequence through base pairing. And when this happens, then you don't have ribosome binding site and you don't have translation. The other component is called an anti-Shan-Delgado sequence. It can base pair with the anti-Shan-Delgado sequence, freeing the Shan-Delgado sequence for ribosome occupancy and translation to proceed. Now, to check whether this is the mechanism operating at the MET-E switch, I generated 3 constructs, one with the Shan-Delgado alone, one with the Shan-Delgado and the anti-Shan-Delgado, which then would generate this kind of base pairing. And the other one has all 3 elements, which you could have this base pairing and you could have this base pairing occurring. And what you see is that with the Shan-Delgado alone, there is very high expression of protein, which means that the ribosome binding site is completely free to bind ribosomes and initiate translation. But when you have the anti-Shan-Delgado and the Shan-Delgado, then we expect this kind of base pairing, which occludes the ribosome binding site, and there is no expression, as you can see here. Introducing the anti-Shan-Delgado restores expression. But when you have a shorter anti-Shan-Delgado, then you have a less restoration than when you have a longer stretch of anti-Shan-Delgado, which corresponds to more or less the full-length MET-E leader. And what this outcome is, or these results are telling me, is that the MET-E switch operates via masking or unmasking of the translation initiation site. Now, the PP2 switch. The PP2 switch is a bit different. First, there is no identifiable Shan-Delgado sequence for PP2. And so the important question here is, how does vitamin B12 regulate the expression of PP2 expression? Now, when you look at the sequence of the PP2 leader, we find that there is an open reading frame upstream of PP2. It is 1 frame shift from the PP2 open reading frame. And I will call this open reading frame UO, that is upstream open reading frame. Now, I thought that this open reading frame might play some role in the regulation of PP2. So I wanted to check first whether it is translated. And so I made a construct where I fused the UO with the LAGZ and check its translation. in the presence or absence of B12. And as you can see here, this U of is translatable, and not only that, it is controlled by vitamin B12. Its expression is controlled by B12, just like the expression of PP2 is controlled by B12. And so now I began to suspect that the translation of the U of might be linked to that of PP2, and that U of might be acting as a relay between the vitamin B12 riboswitch and PP2 expression. Now, when you look closely at the sequence of the U of and the PP2, you find that they share a start-stop overlap. And translational coupling has been known to occur when two genes share this kind of overlap, because the stop codon of the upstream gene and the start codon of the downstream gene share an active site of the ribosome. And so a terminating ribosome can reinitiate translation at that site. Now, to see whether this U of and the PP2 are translationally coupled, I first introduced mutations to disrupt the Chandel-Gano sequence of the U of, or to disrupt the start codon of the U of, or to eliminate this stop and start overlap. And what you see here with this beta-galactosidase activity assay is that disrupting the Chandel-Gano sequence or the start codon or removing the overlap completely eliminates the expression of PP2, which means that indeed PP2 and U of share a translational coupling between them. Now, to further investigate this coupling as a relay for B12 dependent regulation, I generated constructs where I now introduce mutations upstream of the PP2 start codon. That is, to stop the translation of U of before it gets to that stop overlap. And what you see here is that you see attenuation of PP2 when you stop translation of the U of before it gets to that overlap position. But this happens in a distance-dependent manner. The closer you get to the start codon, the less attenuation. But what you also see is that B12 dependent regulation is not lost, which means that these mutations have not been put on the riboswitch itself. Now, what happens when you put stop mutations downstream of the PP2 start, but then eliminate the overlap, is that you allow translation of the U of to read through past the start codon of PP2. And what we see here is the PP2 expression is much more reduced, which implies that the ribosomes here are not finding opportunity to find the PP2 start. And what we can draw from this is that if you have stop codons upstream of the PP2, then terminating ribosomes may slide forward and find the PP2 start and begin translating. But ribosome stop positions downstream of the PP2 will not backtrack to find the start codon of PP2. And what you can see even here more dramatically is that when you just allow translation read-through of the U of much further downstream to a further away stop position, then you don't find expression of PP2 at all. Now, what these results are saying to me is that there is a translational relay that commutes the vitamin B12 dependent regulation of the U of to PP2. And this depends on the translation reinitiation at the stop-start overlap between U of and PP2. So in summary, what we have shown in our work is that RNA leaders are involved in gene expression regulation in nearly 12% of the MTB genome. So regulation by RNA leaders in MTB is more common and may play an even larger role in sensing and adapting to MTB environment during infection than we currently understand. Also, we have shown that there are two vitamin B12 sensing riboswitches in MTB, but they act via two different modes, and that vitamin B12 may be a host signal that induces gene expression regulation in MTB. To conclude, I want to thank my supervisor, Tina, and the contribution of my colleagues, Alex, Peter, and Zaina, and also the contributions of our collaborators and the funders of this project. Thank you.
you to all the speakers in session 2 for very interesting talks. So we're going to move to our discussion session now, with question and answers. I know that we're keeping you from the break, but we're keen to initiate a few more questions in the Q&A. So if you can think of questions, and we'll kick off now with a few, and that would be great. So if all the speakers could put their cameras and microphones on from this session, that would be great. Thank you. Okay, so we'll start with a question from Yogan Pillay. This is to Tim, and it says, it's crazy that it seems that we are only just beginning to understand a bacillus that was identified 140 years ago. Is it lack of tools or lack of trying? Tim, are you happy to take that one?
Yes, yeah. So, yeah, I feel like I'm dialing in for my holiday in Lanzarote. I feel I should be getting back on the plane and getting back to work for the lack of effort on TB. Yeah, no, so I think there's an important point here. I think during the middle of the 20th century, we lost impetus on the research into mycobacteria because they were tricky, but also because we had BCG vaccination and good antibiotics, which seemed to be doing the trick. Then at the end of the 20th century, particularly with HIV and also global disruption, TB seemed to become more important to us. But I think the reality is that, so that's all important, but actually one of the biggest things is that TB is a tricky organism. I mean, it's fascinating. There's always been in the UK a community of scientists who are excited by the biology of tuberculosis, and many of us are members of the Acid Fast Club, which is the sort of core group of people involved in that. But actually, progress was slow. Because the tools weren't adequate for those needs. Progress is always slow with TB. It's a slow-growing organism. We have in our laboratory people doing similar experiments on TB and E. coli. An experiment which takes a week in E. coli will take 3 months in TB. So I would argue that it's not a lack of trying. I think it's the biology of Mycobacterium tuberculosis which makes tough going for all of us. And I'm looking, the other people I can see on my view are all people who've struggled with an organism which takes months to work with, but also the lack of tools. And now we have those tools. And I think the post-genomic era puts us into a position where the progress we've seen with Terry and Dimitrios, the experimental work they've been able to do has been driven by that change in impetus from the advent of the tools that we have available. So it's a bit of both.
Thank you, Tim. And I wonder if I could just be a little bit provocative now and perhaps generate a bit of discussion amongst the panelists and ask the question, when is too much information too much, really? If we have more information about the bacteria, how is that going to be a problem in, say, for instance, we've got so many mutations that we know about and how that informs treatment down the line? I think maybe Gwen might like to say something about that, and also Tim. Tim, do you want to start since you're already there?
Perhaps Gwen might like to say something.
Sorry, my internet cut out at the most perfect point there, Tim. So wait, what did you end with there? Sorry about that.
Just when too much information is too much, really. If you've got lots more mutations and how that might feed into understanding treatment for people into the future, etc.
Yeah, I think I was listening to the beginning of your talk, Tim, and then I dropped out. Sorry, it's really bad timing. But to your response to Q&A, I think something that concerns me as someone who works on antimicrobial resistance more broadly than just on TB is the fact that TB is often seen as this other disease compared to other bacteria, or it's a different bacteria and it causes a different disease. And I think there's something about the language in that is latent infection, like commensal carriage, or what we call commensal carriage in other bacteria, and understanding that. But I think we're moving obviously into a situation where we have huge amounts of data from different patient subsets, and we need to bring that together potentially with mathematical models to better understand what we can do with that data in terms of who we should treat and how we should treat. And from a personal viewpoint, and trying to understand that diversity in the bacterial populations, rather than just thinking about black and white, resistant or not, to do better at subtle prescribing options.
Yeah, I'd add, I mean, I think you can never have too much information. It's just a question of how you manage it. And I think that's important to us. And I think I noticed in one of the questions, somebody asked us about whether we should have lineage-specific ECOFs or cutoffs for our diagnostic tests. And I think that's something 6 months or a year ago, I would have said, no, we don't need those. But the evidence from our work on Protominid and suggests that perhaps we do need to be aware of not necessarily lineage-specific changes, but differences between geographic locations or the strains of isolates which are operating in an individual community. So if you have a scale at one end of the spectrum where you have personalized medicine, I don't think that will work for TB. At the other end of the spectrum, you have the broad brush approach that we have for TB, where everybody gets the same treatment until it goes wrong. I think somewhere between that, we need I can't think of a term which I could invent now for you, but a sort of in-between version of treatment and management of TB patients, which requires that sort of combination. And we're now at the edge of that with the new regimes which are coming through, using the new drugs, bedaquiline, Protominid, linezolid. We have options, and so certainly at a regional or a national level, there may be a need to start thinking about not quite personalizing, having localized rather than
I was going to suggest context specific perhaps, and where the context is either there we go, okay. It's either the patient or I guess the hospital, like the understanding the diversity at the population level can inform what you do about it, and what's the size of that context that matters would be important to understand, right? As you said with the lineage, that was quite interesting.
Yeah, and understanding that evolution. I noticed Francoise's face popped up momentarily a little bit earlier, and that understanding that evolution of resistance and how that becomes fixed in a population is going to be important as we roll out these new regimes and these new drugs.
Dimitrios, you have your hand raised. Would you like to add to the discussion?
Yes, I would just, in addition to what already Tim and Gwen said, I would just like to add that, of course, having big data or a lot of data and mutations especially is quite important, and it will help us actually provide new ideas and also new solutions. But I think one thing we should also focus is that we need also robust phenotypic data, because not all of these mutations will cause a resistance, for example. And a lot of times, like, robust phenotypic data are still missing. So this is something that we should not forget, I think.
Thank you, and thanks for responding to that, panelists. There was a quick question here for Tim from Gabriele Pollara about the MBL assay and how long it takes to do in its current iteration.
Well, it's most likely more a question for you, isn't it, Isabel? So Gabriel, the MBL assay as it stands at the moment is still a very much a research tool, although we use it in diagnostic, we have been using it in specific diagnostic settings on patients to help out when they're difficult to treat. And the reason I say it's more for Isabel to answer is because she's in the process of moving the research-based tool, which takes us about a day or so to do, to a tool which is usable in a diagnostic setting. And perhaps Isabel could answer that a little bit more.
Yeah, I'd say we confidently could give you a result in 2 hours. Okay, thank you. I'll hand over to Sam. I think Sam's going to field a few questions now.
Yeah, I've been keeping an eye on the Q&A, and Terry, you've had quite a lot of interest in your presentation. So I'll kick off with a question from Neil Stoker. He asks, are there particular classes of genes that tend to be regulated by ribose switches? And how much conservation is there with modern organisms?
Thank you very much. That's an excellent question. So to just refer back to the talk that we just gave here, and also to reflect on the work that my colleague Alex has done, where he's trying to fish for mRNA leaders that are involved in gene regulation. And he's finding that some metabolic pathways are enriched for this type of regulation. And we're looking at areas like cell and cellular processes, information pathways, and PE and PPE PE and PPE genes. And these are just areas of metabolism which we think more amenable to regulation via mRNA leaders. But specifically for ribose switches, the full scope is yet unknown, but we think it's great. As to the conservation across species, yes, there is very high conservation across species and model organisms, say, for example, smegmatis and others, between rubber switches of similar class. So if we're talking about rubber switches that sense vitamin B12, then those rubber switches will be well conserved, especially in the portions or the domains that sense and bind that ligand. The expression platform is a little bit more variable, but on the whole, rubber switches for each class are well conserved.
Thank you, and sticking with you, Terry, if I may. Jennifer Steins asks, why is an author translated before the gene an advantage for the efficient translation of the PPE gene?
Thanks again. So the short answer to that is I am not sure why. What I think my work is showing is that the presence of a U-off in front of an annotated gene is playing more of a role of regulating the expression of that downstream gene. We do not know much about what these U-offs do, really. These have been recently discovered. They're quite pervasive in genomes. Unlike before, when we thought that 5' leaders are untranslated portions of genes. So there's still a lot of work to do to discover whether they make an advantage for efficient expression of the downstream gene or not. But I think at this point we can say that they serve some regulatory role.
Thank you. And final question in the chat for you, Terry, from Liz Corbett. Why vitamin B12? What is it about vitamin B12 that means it's been chosen as a regulator of expression in MTB? Do you have a sense of that, or is it still too early to get a handle on that?
Well, I think it deserves a little bit more study. But we know from some reports that there are differential outcomes for TB when people are deficient in B12 as compared to people with B12. And my sense is that there is an interface here between the host and the pathogen when it comes to vitamin B12. Maybe to attempt to guess where such an interface might occur, I would say we think about how B12 might be accessed by the pathogen. And if we think about the pathogen occupying different niches and sometimes or most of the time occupying a granuloma, then you would expect that it would come in contact with vitamin B12 in the host when it's emerging out of a granuloma perhaps, or when it's entering into the bloodstream where you expect to find most vitamin B12 available in the host. So I would say, just to hazard a guess, one of those instances would be emerging out of persistence or latency. Why B12 specifically being chosen as a regulator for gene expression, I can't say at this time.
Thank you. I'm aware of the time and you've got a break coming up. So if I can squeeze in just a personal question and an open question to the panel. We traditionally work with H37RV in our drug discovery work and in our mechanisms of AMR work in the lab. Should we be looking at lineages, different lineages in our research in the wet lab? And if so, which ones? If you can be brief, that would be great.
So perhaps if you don't mind, I'll answer that. I think you're right, we should do. I think it's important to have a starting point, and H37RV is good for that. So what we do in our work is we have a collection of, an extensive collection of carefully characterized clinical isolates from different settings with different drug resistance patterns. And we feel that once we've got some preliminary data with H37RV, then you move to those clinical isolates and select ones which we think are appropriate for the context of the assay that we're doing. So I think that's going to be important. And we do, as a result of our funding through Panacea, which is a EU-funded clinical trials network in Africa, we have a set of isolates which we are prepared to share with people should they want to have access to those, and they include drug-resistant and susceptible strains.
That's a fantastic resource. Thank you, Tim. It's the end of our session. It remains for me to thank the speakers, Tim, Gwen, Dimitrios, and Terry, for fantastic, informative lectures this afternoon. I'll hand back over to Marc, and then after that, you can enjoy your short break. Thank you.
Thank you. So we've got a 30-minute break now. Excellent discussion. Keep the Q&As going. Wonderful presentations. Thanks, everyone. As you can see, we have the agenda here. So we'll see you back in 30 minutes time when we'll, with Beata, we'll explore the exciting world of what's happening with the WHO Pediatric TB Guidance Update. Thank you. See you in 30 minutes. Welcome back, everybody, and it gives me great pleasure to ask Professor Beata Kampmann to lead our next session, Research into Practice, the SHINE trial, the WHO Pediatric TB Guidance Update. Welcome, Beata.
Thank you so much for the organizers for inviting me to share this particular session, which is a particular pleasure to me because it actually addresses TB in children. And it's really wonderful to also have Dr. Anna Torkova present today, who is a long-standing colleague and friend in pediatric infectious diseases and really has what this World TB says, TB Day says, invest in TB. She's heavily invested in TB for children, and she's made an enormous contribution with the SHINE trial and also the updates of the pediatric TB guidance, over which she is going to talk in a minute. So Anna is a clinician, research clinician who works at the Medical Research Council Clinical Trial Unit in London, but she also is a practicing pediatrician. She has driven teaching and research in childhood TB and also HIV for many years, and she's a member of the WHO Guideline Committee, a trial physician in a number of international trials, and I'm really glad that at some point I whet her appetite for actually working on TB and not just on HIV. And so welcome, Anna. We're really delighted to hear all the breaking news that you have to share with us today. And welcome everybody back. We're delighted to see so many of you in this session. Over to Anna.
Good afternoon, everyone. I would like to thank the organizers for this opportunity to give a brief overview of the SHINE trial results and the updated related WHO pediatric TB guidelines. I'm presenting on behalf of the SHINE trial team. TB treatment shortening is one of priority research questions in the end of TB strategy. Shorter duration can improve treatment completion, reduce toxicity, and reduce costs. Historic adult trials showed that non-severe paucibacillary forms of disease could be treated with shorter duration of therapy, which was confirmed recently in post-hoc analysis of the recent TB treatment shortening adult trials. For childhood TB, we know that the majority of children have non-severe paucibacillary TB. SHINE was the first large randomized controlled trial of a TB treatment in children. It's an open-label phase 3 non-inferiority RCT. We aim to determine whether 4 months of TB treatment would be as good as 6 months in children with smear-negative, non-severe, presumably drug-susceptible TB. We aimed to enroll 1,200 children aged under 16 years and randomize 1-to-1 either to receive standard 6-month treatment or 4 months. The follow-up was 72 weeks post-treatment initiation. On the right-hand side, you can see the used drugs, and we use the standard first-line drugs with or without etambutol as per national guidelines. The 4-month arm was shortened by cutting 2 last months in the continuation phase. We use WHO weight band dosing and the new at the time dispersible fixed dose pediatric formulations. Non-severe TB in SHINE was defined as either peripheral lymph node TB or respiratory TB based on chest X-ray findings, which should have had changes confined to one lobe with no cavities, no significant airway obstruction, no complicated pleural effusion, and no miliary TB. Our primary efficacy outcome was a composite unfavorable status by week 72, which included TB treatment failure, TB recurrence, death of any cause, or on treatment loss to follow-up. The trial was powered on the key secondary analysis, which included children adjudicated to have TB at baseline by an independent expert committee blinded to randomized arm, and we assumed that 80% of all children will fall in this category. Adjudication of TB status was based on the adapted NIH 2015 consensus, which was taking into account history of TB contact, TB signs and symptoms, chest X-ray conclusion as the central chest X-ray review by experts blinded to the randomized allocation, microbiology confirmation, and response to treatment at 8 weeks. We enrolled 1,204 children, 602 in each arm, at 5 sites in 4 countries in Africa and India. On the right-hand side, you can see the map and the main inclusion criteria. In terms of baseline characteristics, the median age was 3.5 years, ranging from 2 months to 15 years. 88% of participants came from Africa. 11% of children were living with HIV. In terms of TB forms, 67% had respiratory TB, 29% had a mixed respiratory and peripheral lymph node TB, 3% had peripheral lymph node TB. 14% of participants had microbiologically confirmed TB. Retention and adherence to randomized duration were excellent. 95% of children attended the week 72 visit, and 94% stayed on allocated treatment. Here you can see the main efficacy outcomes. The upper panel presents the results of the primary endpoint in the more modified intention to treat per protocol and intention to treat populations. The lower panel presents the results in children adjudicated to have TB at baseline. You can see the point estimates. These represent the difference in unfavorable status between the arms. And you can see they are around the 0 line, with a right bound of 95% confidence interval, very clear from the pre-specified 6% non-inferiority margin, which confirms a robust non-inferiority of the 4 months treatment duration. Our pre-specified subgroup analysis by HIV status, region, sex, age, weight band, TB forms with and without peripheral lymph node TB, microbiological confirmation and ethambutol use all gave consistent findings. And the safety outcomes were similar in both groups. Health economics analysis at 72 weeks showed that children treated for 4 months had similar health outcomes and reduced healthcare costs compared with those treated for 6 months. Healthcare costs were reduced by, on average, by $17 per child, and if applied on the national scale, this translates into large cost savings. So SHINE found that the 4 month treatment was as good as the standard 6-month treatment for children with smear-negative drug-susceptible non-severe TB, and the results were consistent across all the analyses performed. Reducing the length of treatment could of course make treatment easier for children and families and reduce costs for healthcare systems, which could be redirected to improve TB case finding in children. And you can find the full results in the recently published New England Journal of Medicine paper with a very good editorial there, and also beautifully done quick take animation. I would like to stop on a couple main implementation challenges and solutions. The first one is how to make recognition of non-severe TB easier, and we're very happy to say that the updated Union Diagnostic Chest X-ray Atlas for Pulmonary TB The team who worked on this atlas also took part in the SHINE trial team, and they not only just illustrated typical and non-typical TB changes for childhood TB, but also highlighted the differences between severe and non-severe TB, as shown on the right-hand side in the bottom. And in the infographics, which were designed by Will Elliott in MRC CTU, make pattern recognition easier. There is ongoing work on computer-aided detection for pediatric chest X-rays. We also think that accessible chest X-ray training for healthcare workers will be highly desirable. And of course, investments are needed to improve access to portable digital chest X-rays in the settings where chest X-rays are not available. The second challenge is that smear testing is phased out from the national programs, and SHINE includes children with smear-negative TB. In our trial, 85 children, 7%, were expert positive, and all of them had very low or low semi-quantitative expert results. So what to do if sputum smears are replaced by experts in TB programs? A few studies showed that a smear grade and expert cycle threshold values results are strongly correlated. So therefore, semi-quantitative, negative, low or very low positive expert results or expert CT values of more than 28 can be used as a possible surrogate for smear negative status. We worked closely through the trial with WHO committees, and the results of the SHINE trial was thoroughly reviewed by WHO Guidelines Development Group. We're very happy to say that the updated guidelines on management of TB in children and adolescents have been launched a couple of days ago, and they include a recommendation of a 4-month treatment for non-severe TB. The operational handbook highlights how to implement in practice these recommendations in different types of settings. And finally, I would like to acknowledge SHINE study participants and their families, and our international SHINE trial team, and say big thanks to the sponsor and the funders, and many thanks to you for your attention.
Thanks very much, Anna, for communicating these, potentially game-changing results from the SHINE trial. I can see we have just about 5 minutes for questions. There are a couple of really important questions in the chat here. One relates to the diagnosis of these non-severe cases in the absence of chest X-ray, and maybe you just want to pick that up.
Thanks, Beata, and it's really great to be part of this symposium. Yeah, this year is particularly wonderful for pediatric TB with the new WHO guidelines, new atlas, the SHINE out, and the new SHINE trial was published in the New England Journal of Medicine. So it's quite a few things like, hopefully will help us to tackle the problems. I did paste my answer in the chat. It was very impressive to see how WHO Guidelines Development Group thoroughly considered all different type of settings, and they put very nice practical guidance in their operational handbook. So they do recommend, they did extrapolate recommendations even in the settings where chest X-rays are not available, with some caveats the settings where expert is available or smear testing is available, and also in the settings where neither expert or chest X-rays are not available. Of course, very careful kind of clinical approach and should be exercised. So what they say is that in those settings where nothing is available, is they should be following 2 criteria. So the child has got mild TB symptoms, if you diagnose or think it is TB, nothing is available. The child has been, for example, from TB household contact and has got mild symptoms of TB, not requiring hospital admission. You can try 4 months of treatment. It's actually interesting that it's also it's a strong recommendation for 4-month treatment in non-severe TB. And we are really kind of very reassured because we're seeing quite interesting evidence from the adult trials who also now have got a special interest in non-severe TB group. So I mentioned in the introduction, historic trials, but also there was a meta-analysis on adult data. And quite recently at CROI, there was they presented from study 31 also results on subgroup of adults with non-severe TB who did really wonderfully well with short treatment.
Thanks, Anna. And clearly, if you look at the chest X-ray guide, diagnostic algorithms, and also at the WHO guidelines, I found them a lot more accessible than previous guidelines. So I think there's a lot of detail that can be found for people who are now having to translate this to their practice in the TB control programs. And that's where really the guidance should then be implemented. And maybe people will have to ask you a few more questions if they're in doubt. I know you have to go, and I think I have to switch over to session 3 now, but thanks again for a wonderful presentation and congratulations to the SHINE trial team, and thank you for giving us what looks like a game changer, at least for the non-severe cases, and standing up for childhood TB with all the impact you've had. Thank you very much, Anna.
Thanks, Beata. Thanks, everyone. We're very happy to answer all the questions. You can find our email addresses in the New England Journal of Medicine as a corresponding authors. So yes, please email to us and we can have a conversation.
Thank you. So moving swiftly on, we are now going to welcome you to session 3, which is looking at the impact of COVID-19 on TB. And I'm sure everybody working in the field has been really, really worried about this particular scenario. There have been multiple publications already showing how many cases have been missed and that we have lost a bit of momentum around the notification rates, etc. So I'm really very happy to share this session with my colleague, Professor François Bayou from UCL. And we have 3 exciting speakers to tell us about their observations of the impact of COVID on TB notifications in various settings. And our first speaker is Dr. Rachel Burke, who is a Wellcome Trust clinical PhD fellow, currently based in Malawi with the Wellcome Trust program there, and interested in tuberculosis and HIV co-infection. And if that isn't enough of a co-infection, now we'll also be able to tell us a little bit about the impact of COVID-19 in 3 different countries. Rachel, I'm delighted to see you, and we hand over to you. Thank you.
Hello. Hi, thank you so much for inviting me to give this talk. Really, really pleased to be here. I'm Rachel Burke. I'm a research fellow at the school and an infectious disease registrar. I live and work full-time in Malawi on HIV-associated TB. So I'm really delighted to be talking to you about some of our work on the impact of COVID on TB in Malawi, and the work of other UCL and LSHTM teams also on their work on TB and COVID. So I'm extremely grateful to Pali Khan, Ditman Garetta, and Jose Paredes Sosa for sharing their work with me, so I can share it with you. So to set the scene really briefly, we know that the direct and indirect effects of the COVID-19 pandemic have delivered a body blow to TB diagnosis, treatment, and care. The World TB Report from 2001, which contains data from 2020, shows that for the first time in decades, really for the first time since the HIV crisis, that TB deaths have increased, with 1.5 million deaths in 2020, which is the same as it was in 2017, and TB notifications have dropped by 18%. To shed a little bit more light on this phenomenon, I'm presenting data and analyses from Malawi, Peru, and South Africa. Obviously, these countries have similarities and differences. So Peru and South Africa are upper middle income countries, Malawi's low income. Peru and Malawi have a fairly similar TB incidence, between sort of 100 and 150,000 per 100,000 per year, whereas South Africa has a much higher TB burden. Malawi and South Africa both have fairly substantial burdens of HIV-associated TB, whereas in Peru, HIV is less common. In terms of COVID in these 3 countries, this is data from the Johns Hopkins COVID dashboard, showing weekly COVID cases per 100,000 people from the start of 2020 to the present. The green line is the first COVID case, that's March 2020 for Peru and South Africa, April 2020 for Malawi, and the blue line is the start of the first lockdown in each country. In Peru, lockdown was very strict. South Africa had a reasonably extensive lockdown, but you'll see there's no blue bar in Malawi as we had no substantial restrictions imposed. Obviously, Peru and South Africa had many more COVID cases than Malawi. That might be in part an artifact of access to testing. And each country has had sort of 3 or 4 waves of COVID over 2020 and 2021. Firstly to Blantyre and Malawi. Since 2011, our team in Malawi have had an enhanced TB surveillance programme set up by Liz Corbett. All people registering for TB treatment who consent supply clinical and demographic data, provide a sputum sample, and we geolocate their household. So this is a map of Blantyre on the left, showing the public and private healthcare centres where you can start TB treatment. And on the right are the monthly TB case certification rates from the start of 2016 through to the end of 2020. You can see the background rate of TB notifications has been decreasing from 2016 to the start of 2020. Those are in blue. And in red are the notifications from April 2020 to the end of the year. And that's sort of our COVID time period. So we fitted an interrupted time series to these data and developed a counterfactual no COVID scenario where the pre-COVID trends just continued to work out the estimated number of TB notifications that were missed or delayed in 2020 due to COVID. As a secondary analysis, we split this between men and women, HIV positive and negative, primary healthcare versus a central hospital. So this is our observed monthly TB notifications and the trend with the interruption to the start of COVID time and the counterfactual predictions in red. So we observed just over 1,000 TB notifications from April to the end of 2020. And in the counterfactual no COVID scenario, we would have expected about 1,400 notifications. So that's 333 fewer than expected TB notifications in Blantyre in 2020, which is a relative decline of 24%. That said, the drop-off was steepest at the start of COVID, so in April, May 2020, and had relatively recovered by the end of the year back to sort of expected levels. As a secondary analysis, we compared who was most effective. We found that women were more affected than men, primary healthcare more affected than the central hospital, and HIV negative more than HIV positive. Although that might be an artifact of people living with HIV being more likely to receive care at the central hospital than primary healthcare centres. So moving on to South Africa, this is work by Pali Khan, Dickman Coretta, and their team at ARI. So this analysis considers the Chibisa sub-district in KwaZulu-Natal, where the Africa Health Research Institute, ARI, is based, and it's the site of a long-running demographic surveillance survey. Among other things, it's a region with very high prevalence of lifetime TB. So in this analysis, Dr. Khan and Dr. Coretta used data from the South African National Health Laboratory System to do an analysis of expert ultra tests conducted in 12 healthcare centres in the Chibisa region. The top graph here is trends in the number of experts from 2018 to the end of 2021, and the bottom graph is the positivity rate of the experts. So overall, 41,700 tests were done over these 4 years. You can see in Q2 and Q3 of 2020, a drop in the number of tests performed, with quite a nice corresponding rise in test positivity. But by the end of 2020, the number of experts have picked up again. And Pali also has data here into 2020 showing an increase in the number of experts being conducted. This is the same 2018 to 2020 data on monthly expert tests plotted in a different way. And here you can clearly see the drop in the number of tests in 2020 and the bump in test positivity towards the end of the year. So finally, moving to Peru, and this is work by Jose Luis Paredes and Dave Moore and team. So Jose used national data from Peru to compare TB diagnosis, treatment and care indicators during 2020, i.e. during COVID, and compare them to the previous 3 years. So between 2017 and 2019, there were an average of 28,200 people diagnosed with TB each year. And in 2020, that reduced to 21,400, which is a 24% decrease. These reductions weren't distributed evenly across Peru, with some districts harder hit than others. So Jose was able to reconstruct cascades of care from the national data for how many people with TB symptoms were identified by healthcare providers, how many were examined by a medical professional, how many had a sputum test, and how many were diagnosed with TB. So you can see a big reduction in the number of people with symptoms being identified, a large reduction in the number of people having sputum tests, feeding through to that 24% decrease in the number of people diagnosed with TB. The team was also able to look at some TB care indicators. So of the number of people starting TB treatment, albeit it's a smaller number of people overall, but similar proportions were screened for HIV and diabetes in 2020 as in 2019. But the number of contacts traced per person diagnosed with TB and the number of sputum samples referred for drug sensitivity testing were reduced in 2020. So to finish in with a few reflections to put this together, and I'm afraid I don't think I have anything particularly original or novel to say here, but only to state the obvious and to restate the 2021 Global TB report that COVID and its direct and indirect effects have had a severe impact on TB diagnosis, treatment and care. And that clearly different countries have been impacted differently, both in terms of COVID and on its TB knock-on effects. It's maybe slightly less obvious, but I think it's sadly clear that we are not all in this together in any meaningful sense. We already know that most people with TB around the world are disproportionately likely to either be poor or live in poor countries, or indeed both. And that probably impacts sort of funding and attention given to TB. And now, likewise, we are starting to see that access to COVID vaccines, COVID treatments are extremely unequitable. Hence, Maddie Pye was warning last week that COVID might become a poor country disease. So Malawi, for example, where I live, has no access to novel COVID treatments, and our access to sort of timely, predictable, adequate volume COVID vaccines with a sensible expiry date to be able to plan a national campaign has been abysmal. But I really don't think this needs to be a trade-off between fighting for resources between COVID and TB, but rather everyone, and particularly people who are poor, deserve access to equitable, responsive, adequately resourced health systems, clinics, and to sensible public health measures so that both COVID and TB, and indeed anything else, can be diagnosed and treated. UNAIDS have a nice publication and report with some language about reimagining healthcare post-COVID for COVID and HIV. And I think the same sort of language is really relevant for TB, and frankly, that poor people deserve better. My final reflection on what COVID might have highlighted and what remains relevant for TB is the need, sure, to have funded human resources for health, but also to consider how we protect our healthcare workers from respiratory pathogens. And there might be lessons here from TB and COVID to learn from each other. I know the IRI team have been doing some infection prevention and control work for TB and COVID, so that might be particularly relevant here. And finally, a huge thank you to all the teams who've worked incredibly hard on these analyses. Thank you to Rebecca and Razasoko, who I worked with in the Malawi data, and thank you to everyone for letting me present your work from Peru and South Africa. And thank you so much to the organisers for inviting me to speak today. Thanks, bye.
Thanks so much, Rachel. And I could see you were already ahead on World TB Day 2023. So we'll look forward to how this pans out next year. We will have all the questions for the people on the speakers in this session at the end of it. And I'm delighted to now hand over to Francois to introduce our next speaker. Please keep posting your stuff in. I can see the chat has taken over rather than the Q&A. So we'll try and keep an eye on both. Francois.
Thank you. Now in this session, we'll have Nabila Shaikh, who's perfectly qualified, is in perfect position to continue in this COVID-19 TB session because she worked on TB previously at the London School, and now she's working on COVID-19 epidemiology at Sanofi Pasteur. And the title of the talk will be, content of the talk will be on the effect of COVID-19 pandemic on global BCG coverage and pediatric TB mortality.
Hello, I'll be presenting the impact of COVID-19 on global BCG coverage and pediatric TB mortality on behalf of the team. The WHO recommendation is for BCG to be given as soon as possible after birth, but BCG vaccination policies vary by country, with the map demonstrating the variation in policies. 152 low and middle income countries have a policy for universal neonatal BCG vaccination. Final coverages vary by country, for example, 37% in Somalia and 99% in Ukraine. And overall, about 56% of the BCG eligible population received the vaccine within 1 month and 89% by 3 years of age. Previous modeling suggests reducing delays and increasing BCG coverage could reduce pediatric TB mortality. Over 50% of the global BCG supply is produced in India. In 2018, there were 5 pre-qualified suppliers. 1 has since been delisted. The current annual global demand for BCG is about 300 million doses, of which UNICEF procure 50%. Harris et al. Showed that even a small temporary shortfall may markedly increase pediatric TB deaths. TB-related services have been affected by the COVID pandemic, from case finding to the provision of treatment. Modeling analyses estimate that COVID-19 related disruptions have resulted in an increase in TB incidence and mortality. There have been large reported BCG coverage reductions in some countries, with a 41% decline noted in Pakistan and 52.7% decline noted in sub-Saharan Africa. However, in some countries such as China, there have been no reported disruptions to BCG. At the time of this study, there were no comprehensive data on the impact of COVID-19 on BCG coverage globally, or any estimates on the likely impact on global pediatric TB mortality. Therefore, our aim of this study was to quantify this impact on BCG coverage and estimate pediatric TB mortality. To conduct this analysis, we used a static mathematical model in Microsoft Excel, where we estimated the risk of TB death in the first 15 years of life and any additional TB deaths due to delayed vaccination. The global BCG eligible population was defined as the annual birth cohort in countries where there was a policy of BCG vaccination. For this analysis, we calculated the number of children at risk of TB death by calculating the number of unvaccinated children, and in addition, the number of children vaccinated but with an insufficient immune response. This was multiplied by the weekly individual risk of TB death in unprotected children. We assumed that the BCG coverage reduction was only applied to those born within the disruption period, and the outcome was difference in number of TB deaths in the no disruption scenario, for example, if there was no COVID pandemic, compared to the disruption scenario. We also conducted uncertainty analyses. We estimated the baseline number of TB deaths by 15 years of age in countries with universal BCG vaccine policy. The data we collated to estimate this was the births and childhood deaths, TB incidence estimates, childhood mortality and risk of TB, BCG final coverage estimates, immunization timings, and vaccine efficacy. At the time of this study, there was no global unified record on BCG disruption. Therefore, we used peer-reviewed papers, newspaper articles, conversations with experts, and confidential sources to collect this data. At the time of the study, there was limited data on the duration of disruption, with varying 3 to 6 months estimates from various settings. Therefore, we used both 3 and 6 months disruption durations in this analysis. For the BCG country-specific disruption data, we found data for 28 countries, and these 28 countries cover about 61% of the global BCG-eligible population. Where we were unable to find BCG-specific disruption data, we collated data instead on the interruption of general immunizations as a proxy. For this, we found data in 2 countries which cover about 4% of the global BCG-eligible population. And some qualitative studies noted that there was limited disruption of BCG when it was given at birth in facilities. And UNICEF data suggested that about 23% of all births took place outside of health facilities. Where no country data on disruption to BCG or general immunization services could be found, we used a weighted average of coverage from countries where we did have data. And the weight was proportional to the global BCG vaccines delivered by each country in 2019. And this analysis, we estimated that the global reduction to BCG coverage due to COVID disruptions was 25%. So we developed 2 key scenarios. One was a disruption period of 3 months, and one was a disruption period of 6 months. We also assumed multiple scenarios for catch-ups, ranging from no catch-up at all to catch up to 50% of the deficit within 3 months after the end of the disruption period, 6 months after the end of the disruption period. And we also used MMR as a proxy for potential for vaccine to be co-delivered with another immunization, and MMR is usually delivered at a lower rate than BCG. So this is just a breakdown of our scenarios. This is scenario A, with the disruption period lasted for 3 months. In scenario A1, there was no catch-up. In scenario A2, there was a catch-up to 50% of the deficit within 3 months after the end of the disruption period. Catch up to 100% within 3 months, catch up to 50% of the deficit within 6 months, catch up to 100% of the deficit in 6 months, and then catch up to 50% of the deficit timed with MMR. And as you can see from the graph, the MMR delivery is a little bit more delayed compared to BCG baseline. and catch up to 100% of the deficit timed with MMR. And similarly, we use the same catch-up scenarios, but instead with the disruption period of 6 months. So it's the same catch-up scenarios as we've seen in the previous slides. So this is a summary of our model scenarios with a disruption period lasting from 3 to 6 months with various levels of catch-up. And as you can see, the magnitude of disruption was greater for the scenarios with a 6-month disruption period. So these are some of our results. This graph shows the percentage increase in TB deaths in the first 15 years of life compared to the baseline scenario. So the baseline number of deaths, if there was no disruption, was about 194,000. In the best case scenario, we saw a 0.5% increase in the additional number of deaths, and in the worst case scenario, 17% additional deaths. So in the best case scenario was scenario A3, so a disruption of only 3 months and catch up to 100% of deficit within 3 months after the end of the disruption period. And the worst case scenario was a disruption for 6 months with no catch up. And these results in absolute values, there was 886 additional deaths in the best case scenario, so limited disruption with catch up immediately. And in the worst case was disruption for 6 months with no catch up. We found the length of initial disruption to be an important factor in determining the magnitude of additional deaths. So for example, these are the scenarios considering 3-month disruption versus scenarios considering 6-month disruption. The level of catch-up was also determined to be very important in determining the magnitude of additional deaths. These are the scenarios with 50% catch-up versus the scenarios, the same scenario, but with 100% catch-up instead, delivered in the same time period. We found that the time taken for catch-up was less important. So for example, in the pink, you can see scenario A2, which is catch up to 50% of the deficit within 3 months. Scenario A4 is catch up to 50% of the deficit within 6 months. And scenario A6, which is the catch up to 50% of the deficit, which is timed with MMR, which is more of a delayed schedule. And you can see that there is a little difference between the magnitude of additional TB deaths. And these are the same scenarios, but with the 6 months duration disruption instead. So in summary, where the data was available, we found that the disruption period ranged from 0 to 100% for a period of between 3 to 6 months. And we now know that the disruption period varied substantially by country and region. The model suggested BCG disruption may increase childhood TB mortality by 0.5 to 17%. We found a shorter interruption period with a larger catch-up campaign will minimize excess TB deaths, and action is needed to minimize BCG disruptions due to COVID and catch-up. Catch-up timed with MMR or other immunizations may be a feasible way to deliver vaccines without additional resources. However, we did find in our data that MMR was also disrupted. Some limitations of our study: limited data was available on BCG disruptions at the time of this study. We were unable to find data for countries contributing to about 35% of global BCG coverage. We didn't consider any unrelated disruptions or interruptions in 2020, for example, due to war stockouts. And we didn't consider that some countries had extended or recurrent lockdowns. There also may have been some localized interruptions that were not included in country level estimates. We also know that BCG may have other direct and non-specific effects. Therefore, the impact on additional pediatric deaths may be greater than we've estimated here. And our wide confidence intervals are largely driven due to the uncertainty ranges for vaccine effectiveness, vaccine efficacy against TB death. So in conclusion, COVID-19 disruptions to BCG coverage could increase childhood TB pediatric mortality, and action is urgently needed to stop ongoing disruption and catch-up. If feasible, the BCG vaccination can be given with an alternative immunization to prevent additional stress on the immunization program. Better BCG coverage data now will lead to better impact estimates, so we are now able to conduct perhaps a retrospective analysis, and mitigation measures against disruptions should be prioritized as soon as possible. BCG market supply is still delicate, and action is urgently needed to stop ongoing disruptions to BCG. We'd like to acknowledge all individuals that provided input into this analysis and Parfa Roy for the base model. And these are just some references that we use for the study. Thank you.
Thanks very much, Nabila. That was excellent, and really other critical issue, what I would call is COVID collateral. It's now my pleasure to introduce Dr. Jayne Sutherland, who is a colleague of mine and associate professor at the London School of Hygiene and Tropical Medicine, and the lead for our tuberculosis immunology research here at the MRC unit in Gambia, where she leads a large TB case contact platform. Jayne is an immunologist and will talk to us about the influence of the current and recent COVID on the performance of novel TB diagnostics. And this is an area in which Jayne has worked very extensively over the last 15 years or so. Jayne, take it away.
Thank you very much for this wonderful talk. And I think now we're going to have some questions. So the first question is probably a question to both Rachel and Nabila. It's been on the chat, and essentially either of you see also some possible positive effects of COVID mitigation. Let's say, obviously we've heard a lot about the negatives, but do you think there might have been, let's say, lesser transmission possibly due to some mitigation measures? So I'm not sure who wants to answer that, because it's really a question for both. I think Rachel and Nabila. What order you want to? Maybe Rachel first?
Yeah, I'm happy to answer, but I'd also love to hear from Nabila as well. I mean, I think it's hard to know. I think through 2020, which is when our sort of Malawi initial analysis ended, I think through 2020, that's probably all just disruption to services and people not accessing care. I think when you sort of get into 2021 and 2022, it's much harder to know what your sort of, had there been no COVID, what the TB incidence would have been, and therefore what this gap is. I mean, that sort of, 18% drop in notifications through 2020 is not due to reduced incidence. And because we did try to think about extending our Malawi analysis through 2021 and 2022, and we really struggled to think, what sort of functions and how we could come up with some sort of sensible counterfactual. And I spoke to some people at TB Modelling Centre, and we never got that far with it. My opinion is that the negatives are going to outweigh the positives quite drastically so, in terms of just massive disruption to care, possible disruption to HIV care, access to diagnostics, and then sort of economic shocks and worsening poverty and whatnot. But I don't know. I wonder if Nabila's got anything that's worth saying.
Yeah, no, I completely agree.
I'm sure you do.
I completely agree with your points. I think if we looked at influenza as a sort of proxy for interruptions in transmission for COVID, there's been a big drop in influenza transmission over the last 2 years. But I definitely agree with all of your points that the negatives will probably outweigh the positives. I know many teams are looking into the point as well and trying to understand whether the drop in notifications is due to incidents. Hopefully we'll be able to get some more clarity, but yeah, no, I completely agree with you, Rachel.
Well, thank you very much. Well, we're hoping for some.
There is a question for Rachel, which also, I can also put to Jayne and to some degree, and that relates to maybe also opportunities that have come out of the COVID pandemic. So there is a question here from Grania, asking, do you think the investment in lab systems and machines like Expert and chest X-ray for COVID diagnosis can support the catch-up required for COVID? I think it's really important to connect these investments. And by the same token, Jayne, you're very actively involved in trying to get to point-of-care diagnostics for TB. And do you see an opportunity that, given the fact that we had COVID diagnostics developed in record time, how does that relate to the TB space? So maybe Rachel first.
Yeah, thank you. I mean, I think, like I said, I don't think it should be like put in this big like either or. And I've seen lots of things from World TB Day about this, like, so much money for COVID and no money for TB. And on the one level, of course, I see where people are saying that, and of course we want more money for TB. Because the driver is that like lots of rich people get COVID, and insofar as TB gets neglected because it's often poor people and people in poor countries, of course that's not fair. But I don't think it's particularly helpful to be saying, my disease is more important than everybody else's, when what people need is sort of a holistic assessment of all their healthcare needs. So if they've got TB, they need a TB diagnosis. If they've got sort of severe COVID, they probably need a COVID, they need a COVID diagnosis. So I think if we take a person-centered approach and see what people need, then that's just a bit more helpful than shouting about our various diseases. But absolutely, I mean, I think then you say, well, what people need quality assured diagnostics for, primary care and for like evidence-based screening that's going to benefit people. I'm not sure where we are with that for either disease, quite frankly. But chest X-rays on the WHO essential diagnostic list, so people should have access to chest X-ray. Molecular diagnostics for TB, for HIV viral load, for COVID are going to be helpful to lots of people. And if COVID can help leverage us get those so that everyone benefits, then that would be absolutely fantastic. And Jayne highlighted these huge big gaps that we have in the TB cascade of care. So if we can get sort of more access to X-ray, more access to expert through some sort of COVID thing, then fantastic.
Jayne, do you want to pick this up on diagnostic side? Yeah, great.
I'll just add that I'm hoping, the one benefit of COVID in terms of TB tests and vaccines is that the technology is driven forward. And so what we want is to be able to access these, but access it so they're field friendly. And currently, a lot of these tests aren't yet at the stage where they're field friendly, but I feel like we're on the cusp. There's the technology is there, and we just need the investment to put it on a single cartridge that can be run and do all these multi-pathogen tests and the host response as well. But the host response is very good in terms of screening, so it remains to be seen what will happen when we analyze the full cohort. But certainly that's going to help in terms of streamlining and giving people access to further TB diagnostics.
So Liz Corbett is asking a question in the same vein. Why do you think that Cepheid has done so much better than competitors? Does anyone want to risk a take on that situation?
We're sort of linking with a company, Quantum Diagnostics, who are a similar sort of technology, but they've moved it forward, so it is portable, it's handheld, you can take it to the field. Whereas Cepheid haven't quite done that, or they've done it, but then they've retracted that technology. So I do think there's other companies there, but the Cepheid, certainly the fact that it's so interchangeable with the programs on the machine, they've really worked that software out very well. And I don't know enough about the other quantum diagnostics to know how easy or difficult it is, but certainly they went into COVID and sort of have neglected the TB field. So we're sort of hoping that they'll come back to the TB field.
Yeah, maybe the platform that existed so widely was really also really good. Nabila, I'm really interested in your modeling of the vaccine delivery during the times of COVID, because we know, and it was particularly interesting since you picked the comparison or the potential to give it together with the measles vaccine. I mean, you're probably aware that there's a huge lack in measles vaccination during the COVID time as well. Do you think the BCG question is a bit different from what happened with the other vaccines? Do you have a take on sort of how that might pan out in the wider vaccination field? Because it's a neonatal vaccine. There was also a question, when is the BCG vaccine recommended? Just to answer that briefly, the general recommendation in endemic settings is to give it at birth or close to birth as possible. And there are different issues with accessing services at birth compared to later on. Do you just want to comment on that?
Yes, so I think multiple different things to talk about. Yeah, just to go to the question in the chat. So yes, the vaccine is recommended to be given at birth, but as you just mentioned, there are some delays due to the practicalities of giving neonatal vaccinations and immunization programs. So some of the data I presented was just when the maximum coverage is reached, which is around 3 years of age. Definitely, we know that there were big impacts to other vaccinations and programs. I think the idea of the study was just to show that some catch-up is required as soon as possible. And if the catch-up is slightly delayed, so for example, we use the MMR program as a proxy because it's given at a slightly delayed schedule, it still has a bigger impact than not offering any catch-up at all.
Okay, any other questions? Well, on the issue of BCG, Francois, I think you had a question for Nabila and maybe everybody else.
Well, I think it's been partly so, but in the very early stages of the pandemic, there were some hopes that BCG might have some potential positive impact on COVID. So do you think there's still any kind of evidence for that?
A lot of the evidence at the start seemed to be quite promising, but I've not seen anything recently to suggest that BCG has a great impact on COVID, but we are seeing I've seen some papers on other vaccinations such as polio potentially having a positive impact on COVID. I think with BCG, we know that it has a positive impact on wider pediatric health as well. I think we can't rule it out. I think it'd be very interesting to study it. Yeah.
The clinical trials that have been done have not shown any advantage in the context of BCG revaccination, just to make that clear. Okay, so Jayne, do you think that we need to assess all these new technologies with everything new virus that comes into town?
No, I don't think so. I think, I think at the moment what we can see is that the performance didn't change much with COVID. I don't know, like possibly, but I think some of the data. So Simon's paper, which is really lovely, but the way they detected the pathogens is different to how we've been detecting the pathogens. So there's certainly a possibility that other pathogens will influence the test results, but I wouldn't go so far as saying we need to test it against everything, because at the moment the performance is really good, regardless of what and we don't know. I'm sure that when we analyze the 26-plex pathogens we're going to look at, I'm sure there's lots of other co-infections we're not aware of, and yet that performance really stands out. So yeah,
I think it's really reassuring, because there's so many other viruses going around every winter, and if we had to have tests specific for what is the new kid on the block, we'd be on a hiding to nothing with TB diagnostics. Right, we're running out of time now, and I need to hand back to Marc Lipman. There is a question from Liz Corbett in here, or just looking for people who have experience with post-TB clubs or something like that. How do we help patients to live well with damaged lungs? This relates a little bit to talks we've heard earlier. So if anyone has any suggestions there or so, you could email Liz directly, please. So Marc, I'm handing over back to you, and you can close the session and hand us over to the UCSF part of it. Thanks very much, everybody, for their contributions, and our speakers for excellent presentations.
Yeah, thank you. Really, really excellent and thoroughly enjoyable and lively discussion. So well chaired, Beata, as well. Now it gives me great pleasure to hand over to Finn McQuaid, one of the directors of the School of Hygiene TB Centre. Finn is going to summarize what we've heard today, and then we'll move on to the next session. Finn.
Thank you, Marc. And thank you, first of all, to all of the speakers for such really interesting talks that have provoked a lot of thought. It's been great to see such amazing research going on at UCL and the London School, and indeed around the world. Thank you in particular, I think, to Rahima for sharing her experience with us at the opening. It should go without saying that it's vital we be grounded in the patient perspective in all the research that we do. So we really valued hearing from her today. And I think understanding the longer term consequences of TB beyond treatment completion, centered on what that means for the individual concerned, is clearly a vital area of study going forward. It was also great to hear about the new WHO pediatric TB guidance. I think COVID-19 has really brought into the public spotlight the need for an equitable approach to infectious disease care and control, where children remain a key marginalized group for TB. I'd also like to thank all of those who've worked so hard to pull this together, particularly Marc, yourself, Anna, Hanif, Tina, Eloise, Neil, Ryan, Sean, Essen, Maria, Anna, and Alicia. And thank you also to you, the audience, for joining us. I hope you found it as engaging and interesting as I have. As Marc said, we will now be moving on to the last session on clinical trials, and in the tradition of handing the World TB Day baton over to UCSF, this will be a joint session with those in California. After this session, the UCL London School Symposium will be closing, but the UCSF meeting will continue for those of you who are able to join. You can find a link in the program, and Marc will outline this more in a minute. And then lastly, in closing, I think it's right that we also acknowledge the awful situation in Ukraine currently. As with COVID-19, which we have just heard has had such terrible consequences for TB, so too with ongoing conflict in Ukraine, in Tigray, in Yemen, Afghanistan, Myanmar, and elsewhere. Not only the devastating effect of these conflicts on lives and livelihoods, but on top of that, the resurgence of preventable diseases with long-term consequences, such as TB, as a result of these conflicts. And it's abhorrent that those in power in Russia and elsewhere countenance these actions, which are so earth-shattering for the most vulnerable and marginalized amongst us. I think that just really highlights for me more than ever the importance of all of the research and efforts that we've heard about here and that many of you are involved in to end TB. Thank you. Marc, over to you.
Thank you, Finn. Very powerful words at the end of our session, and we had a very powerful presentation, as Finn said at the start of it. The information how to join the UCSF meeting is now in the chat. You also will have received an email on it, so do try and connect to that for what will be an excellent session. And I would just finally like to add my thanks to those of Finn, to say to the organizing teams, both the scientific and the administrative and the newfound technical skills from people within UCL-TB and the School of Hygiene. It's been so appreciated, this, and thank you for making this a really excellent meeting. We still have to continue, and we've still got one more session, so I'll see you on the other side, so to speak, at the UCSF event. Thank you. Have a good afternoon, evening, or morning, wherever you are in the world. Thinking of you.